So good morning again. This is the last day of the hackathon, and today is dedicated to integration of networks in closed-loop controllers, robots, and virtual brains. So the opening lecture will be given by Gidio Falottico on cerebellar networks in robots.
Okay, good morning everybody. Today we'll talk about the cerebellar networks for robots and why we need this kind of networks in order to improve the robot control. We start from the fact that the control of the movement in robot and humans is very difficult, and we try to rely on the very special capabilities that humans have in the motion control, and these capabilities are related to different control issues. For example, if we look at the goalkeeper or the butler, you can see that in order to anticipate the motion of the target that is the ball, they need several special capabilities, like the rejection of the disturbance, because they, in a way, they are dealing with the motion of their body, but also of other device, like for the butler, and what they need is also to have other issue like the capability to predict what is going on in the environment. And for this reason, one of the main specific capability that they have is the prediction of the target motion. Of course, the goal of this is to try to replicate this in robots, and as you can imagine, this is not easy because the perception of the environment is very difficult if we think that it has been done using artificial devices, and also the control of the robot itself. If we consider a humanoid like this one in the movie, consider that it has very high number of degree of freedom, and at the same time has to control the different joint, but also to understand what is going on in the environment. So we try to rely on neuroscientific evidences and models in order to improve the capabilities of the robot. Starting from neuroscience, we see that actually, neuroscientists try to understand the brain like an encoding-decoding machine, where actually the encoding is how actually the stimulus can be perceived by the brain and transformed in spike, for example. And we want also to decode, to understand what happens when the brain sends command to the body, and what this decoding, what these responses tell us about the stimulus that the robot or the human can have. An example which is very well known is the encoding of the stimulus, for example, in the visual cortex, where they found evidence show that actually there is a relation between, for example, the orientation angle of the object perceived in the environment and the spiking pattern that you can record in the visual cortex. In the same way, this is not only related to the perception, but also to motor cortex. So recording in motor cortex show that there is this pattern recognition also there. Actually, there is this encoding of the motion and specific, for example, direction of the movement are encoded by the neurons. But if we see the encoding only as a descriptive way to understand the brain, of course, this is not going to work if we want to apply this to robots, because the same neuron can encode different parameters that, for example, for the motion or for the perception. And what we would like to understand is that the mechanisms that are behind the neural activity more than what they encode. So we need a kind of descriptive model that, for example, can allow to control the arm or the body in general and predict what will happen in the environment and on the body itself. So several theories have been applied for this. You see here, this is an example of Todorov, which studied this interaction between the body and the brain using an optimal feedback control method. And actually, he simulated the body with this simple function, which is actually used to create a causal relation between the inputs to, for example, in this case, the arm, and what will be the feedback that will be received by the brain. Of course, the focus of this model, as I said, is the flow from the cortex output that goes to the spinal cord and can control the muscle force production. We should also consider that this is applied to a complex structure like, for example, the arm, which is a multi-joint mechanism in order that is needed to reach the target point. So first they hypothesized that how the motor cortex might be causing this movement, and then they create a correlation that could explain the emerging property of this causal flow. What is the problem of using a simple feedback-based model? Actually, there are three main issues related to this. The first one are the sensorimotor delays, because actually our visual system, but also the proprioceptive one, has a delay. So this creates instability, as we will see in the control of the arm in this case. So actually, this is the control scheme proposed by Todorov, and starting from a task selection where we actually say what we want to achieve, there is this closed-loop control which is used in order to make the eye or the arm reaching a fixed point, and he modeled this using an optimal feedback control policies because, for example, this allows to overcome problems like the delays or the redundancies that you can have in the system. So if we consider, in fact, what is a simple feedback delay control loop, we see that when we introduce delays or when we change, as in this case, the feedback gain, we have different results according for what concerns the output of the controlled object that can be an arm or the eye itself. I show you here what happens in a very simple MATLAB script. For example, when you have this simple system, we consider a function that can replicate the dynamics of the eye, and we use, I don't know if it's visible, a simple, yes, should be visible. We introduce as a proportional controller, which is, we start with 1 here, and we have no delay. We see that the system is stable, can reach the position and keep it. But as soon as we increase the delay here, our system dynamics completely changes and starts to become more unstable. Of course, if we further increase it, the system becomes more oscillating, and for very high values, it becomes not controllable. So this is the results of a very simple experiment that we could have for the control of a simple system like here, a closed-loop control, where we just change the delay. So if we think that in human we have different delays according to the sensory incoming stimulus, you can imagine what is the amount of information and also the needed control strategy that can help in maintain a stable control for the dynamic object that we want to move in space. So I showed you the different what happens when we change this parameter, for example, when we increase the delay. So the question is how the brain can deal with the delay in the sensory feedback. Actually, neuroscience made two hypotheses for this that were studied in detail. The first one was the equilibrium point control, where actually they hypothesized that the brain just sends a simple command to the arm or to the eye or the system that he wants to control, and the real control part is done by a complex spring-like muscle control. So this means that the brain has only a high level visibility of what is going on, so it just sends the trajectory in space, and the real control of the arm is done by the capability of the muscle itself. The other theory was the internal model-based control, where actually they say that there are complex brain commands, and the control done by the muscle is a simple control. In order to verify this, actually they made an experiment like this one, where a human was, for example, or a monkey was attached to a robotic arm, and they did this experiment where they have to reach a target under perturbation or maintain the position when there is an external perturbation. What they found out in this study is that actually in the case where the monkey was asked to keep the position in space when we applied here, for example, a step torque perturbation, a fixed one, the MEG activity of the muscle that you can see here actually was evaluated according to the delays in time to see what happens according to the feedback time that arrived to the brain in the cortex, for example. So what they found out is that the response is observed during the long latency and voluntary epoch. As you can see here, the main part of the response is here when also the visual feedback is active. This means that it's the brain that is controlling the arm in this case, and it's not a property of the muscle that allows to keep the position in space. So we should consider that the brain receives a large amount of sensory projection, and it is not always easy to understand which area is responsible for what kind for the behavior, since there are several connections inside, and some of them are still not clear in the brain. So also here again they try to measure in different brain areas to understand what happens during the task of keeping a fixed position while an external disturbance is applied. And what they found out, and as you can see here in the different responses of the brain areas, is that a lot of them fire vigorously when the mechanical perturbation is applied. So this means that actually arcs against the involvement of the equilibrium point hypothesis, which was not valuable for explaining this kind of behavior. If we try to make an history of this, actually the equilibrium point theory was proposed by Leitasch in 1993, and this, as I said, they say that it was the spring property of the muscle that allowed to reach a point or to keep a fixed position. But in 1996, Gomi and Cavato, with this paper, demonstrate that this theory was implausible. Actually, what they have done was an experiment, another experiment with a manipulandum, where actually they proved that the equilibrium point trajectory that was estimated from the measured stiffness and the generated torque, and actually having a look at the profile, the velocity profile that was generated by the arm, they saw that this was not the same that was sent to from the brain. So this means that the equilibrium point theory is not plausible for explaining the control of the arm. So from this one in 1998, Wolpen, Miel, and Cavato proposed this new hypothesis related to internal model, the presence of this model inside the brain that are built inside the brain that in a way encode what is going on in the body and also in the interaction with the environment. And in 1999, Cavato proposed what is called feedback error learning paradigm, where actually this was used in robots for the control. So actually, starting from this theory proposed by Cavato, we can recognize two types of internal model. The first one is the inverse model, that the role of the inverse model is to take the desired state trajectory as an input and produce the motor commands that are needed to move the body accordingly. So actually, we add this to our basic feedback control because this is needed in order to overcome all the issues related to redundancy and to delay that we can have in the system. Another type of internal model is the forward model that takes the copy of the muscle command that the body receives from the motor center and generates the prediction of the current and future state of the body. We need also an integrator region that should continuously integrate the prediction of the internal model with the respective delayed feedback that are coming from the body and produce an estimation of the most likely body state. So, in summary, the sensorimotor control with delays and variability taking into account the forward and inverse model that we have here, where you see motor command and generator, we have this inverse model, which is composed in this case of a pure feedback plus the feed-forward controller that I showed you before. And also we have a forward model that sends input to the integrator in order to update our feed-forward controller. What is the role of all these models? The idea is that we need to compensate for the delay. So what you will see is that if we consider the behavior of, for example, when we move an arm in space, what you have is a sensory prediction that is needed in order to accomplish the task. So the question is, and what was proposed also by Cavato, what is the brain region that actually allows to generate these internal models and improve the control for human? And it has been demonstrated in several tasks that this brain region is the cerebellum. So what we need is a capability of predicting the movement, but at the same time to learn what is going on in the environment in order to store an internal model that could be reactive and allow to have a fine control of the body. We started with an example, which is the Saccade movement, as an evidence of the cerebellum as a forward model. And Saccades are ballistic eye movement, you may know, that are used in order to shift the gaze from one point to another. Usually, these kind of movements are very fast. They have a very high acceleration and velocity, and during this motion, the sensory feedback is completely absent. It means that there is no vision active during the motion itself. So these are, for example, Saccade movement, of course, are very fast. You see just the final part of the motion. And also, neuroscience study what happens, for example, where you have impairment, and that the saccade motion becomes, for example, not accurate as in normal subjects. So if we consider saccade movement where actually there is no feedback active, means that our very nice control schema lose a very important part, which is that one related to the delay and the sensory system information. It is good that we lose the delay, but at the same time we lose also all the information related to the feedback, and so we started thinking how this could be modeled in order to reproduce, for example, this behavior in robots. So looking at the literature, several aspects are presented for the saccades. One of these is, for example, that there is high variability, which depends also on the importance and the reward of the stimulus itself. So there are evidence that demonstrate actually that saccades have different speed and different acceleration according to the rewards that the stimulus provides. So if we consider what happens during a saccadic eye movement, these two graphs actually show, for example, the amplitude of the saccade with this variability, which is dependent, as I said, on the importance that we give to that stimulus. And also this, even if, as you see, both are very accurate and reach the same point, but what happens, of course, is the velocity that the eye can reach for reaching the same point. What happens when there are impairments, cerebellar impairment, is that actually there is this variability because, as I said, when we have a target which is more rewarding, our velocity is higher. And what happens is actually that we lose the accuracy, as you can see here, because there is not any more refined control which is provided by the action of the cerebellum. So if we try to summarize what we have seen so far, the goal and the strength of the motor command that initiates the movement depends a lot on the value that we give to that point. So there is this variability that is possible to see also because there is this difference in the motion related to the velocity and the acceleration. So what was estimated was that the cerebellum is involved in the prediction of the state of the eye during the saccade in order to make it reaching a goal. If we simplify a lot the scheme that is used in order to control a saccade, when a stimulus appears in our retina, what happens is that the stimulus is directly sent to the superior colliculus, which has this logarithmic map. And what happens is that we want to move from one point to the other inside starting from what is the output of the superior colliculus, and this is directly sent to the motor neuron through the bus generators. But there is also the action of the cerebellum, which is actually depending on the efferent copy of the bus generators that allows to reach the target in a very accurate way, because if we cut this part related to the action of the cerebellum, we will have saccade which overshoots the target. So thinking about a computational circuit of the cerebellum, we started from that one proposed by Dean and colleagues in 2010, where this cerebellum is as this, they replicate what is called the microcircuit, which is composed by MOSI fiber, parallel fiber, Purkinje cell, and clamping fiber. And actually what they do, the idea is that when the cerebellum receives input that goes through the MOSI fiber, they generate a high-dimensional representation by means of the granular layer, and the output is then sent to the Purkinje cell, where actually the learning happens and they can control the eye. There have been experiments on monkeys also for this in order to understand what happens. Recordings were done in the 6th and 7th lobe of the cerebellar vermis in order to understand what is the behavior of the Purkinje cell in the specific, in order to understand what is the real contribution of this cell for the control of the eye during the saccade. One of the first studies was this one from Otsuka and Noda in 1995, which we can consider not really successful because actually they try to create a correlation between the kinematics of the motion of the eye that you can see here, for example, different target. They try to reach a target at 30 degrees, 20, and 10, and they try to correlate this with the discharge rate of the Purkinje cell. But actually they were not able to find any correlation between this. What is interesting here is that in order to make this correlation, they sorted the output of the Purkinje cell based on the simple spike that happened inside the Purkinje cell layer. So in another study in 2015, actually, what happened is that Herzfeld and colleague tried to change the paradigm in the sense that what they did a different sorting for what concerned the Purkinje cell. In this case, they considered the complex spike, and the sorting was done based on this. And what they hypothesized is this. Actually, this connection between, starting from the inferior olive that project through to the Purkinje cell, and different Purkinje cells which have pause and burst activity give their output to the caudal fastigial nucleus. So starting from this, they try to make now again a correlation between what happened to the eye kinematics during the saccade and the output of the Purkinje cell. And they were able to find a correlation between this. You see here, for example, the change in the population response according to 3 different target saccade at 10, 12, and 15 degrees. And they also clearly show actually the capability to predict, the predicted capability of the Purkinje cell that actually anticipate the kinematic motion of the eye. So starting from this recording, we try to make a model from this where actually we want to have, of course, a closed loop, even if, as I said, there are problems because vision is not active during a saccade. And for this reason, we had to deal with this problem, but we used, for example, plasticity in order to learn the task by changing, for example, the PFPC activity in the cerebellum. This was our scheme that we used for the control of the robotic eye. We implemented the cerebellar model as an ecostate network. Here the model is a read-based model. And from this ecostate network, we have a final layer of Purkinje cell, which have both burst and pause activity. And we send this to a simplified bus generator, which try to replicate the activity of the brainstem, and we send the output to the eye. We perform a saccadic adaptation task because we want to show the capability of the cerebellum to learn and to adapt according to the position of the target. So we used this task because actually it was possible to rely not on the magnitude of the error, but just on the sign. This means that our algorithm just wants to understand if we overshoot or we are under the current position of the target. This is the only information needed in order to, with learning, try to improve the accuracy in reaching the target. And in the end, we were able to have the same results that were showed by Hertzfeld in 2010. Actually, you can see here different amplitude of the, for different amplitude of the eye movement, for the Saccade, for 20, 16, and 12 degrees. What is the Purkinje cell activity response, and also, again, the capability of the Purkinje activity to anticipate the kinematic motion of the eye, in this case to anticipate the eye speed, which is the red part here. We also we studied the correlation, finding results which are very close to those one produced by Hertzfeld in 2010. We are now doing an ongoing work is to have a Saccade spiking cerebellar model. We are using the model from Egidio and colleagues. It's a simplified model. Actually, we used, for what concerns the MOSI fiber, we have different input that we use there. And actually, this for what concerns, the inferior olive, there we have, also for that we have a simplified model. We are not using the current model of the inferior olive. We also simplified the activity of the Purkinje cell because we simulate the pose activity by just put a negative value for that kind of Purkinje cell. We don't have a real pose activity for this. But the initial results are quite good in the sense that the model is able to learn and to control the eye in space. Here you can see, for example, for a single target that we try to reach over different trial, actually the error is decreasing. It means that the model is able to learn and to control the eye after, for example, 30 or 40 trials. We also tried with different targets to see what happens, and as you see, the error becomes between 1 and 2 degrees at the end when we have after 3,000 trials. So we want also to use this to understand if we can use the same learning rule to explain the movement adaptation, and for this reason we try to apply our cerebellar model also to other eye movement like the vestibular ocular reflex. As you know, the vestibular ocular reflex is that ocular motion that happens when, for example, you move the head and the eye move in the opposite direction in order to stabilize the visual input on the retina. So what we need is, of course, an internal model of the dynamic of the eye in the cerebellum that could, in a way, ensure that when the head is moving, the motion of the eye is fast and can balance, and counter-rotating the eye can keep the image stable on the retina. So we start from, if we start from a simple feedback controller where we have a brainstem at the beginning which sends output to oculomotor plant, actually what we see that is that there could be variable plant dynamics but also variability in the sensory feedback. And again, as we have done for the Saccades, we introduced a cerebellar modules that can help, for example, in improving the accuracy and the speed of the eye in order to really balance and counteract the motion of the head. So, as you may know, there are two different strategies that could be used in order to achieve this control with the cerebellum. One was proposed by Cavato, and the other one was proposed by Dean and Porrill. One is called the recurrent architecture, which is the red path, where actually the cerebellum is used as a forward model which changes the input trajectory to the brainstem. In the other case, the cerebellum in the feedback error learning paradigm, the cerebellum is used as an inverse model, and the output cerebellum is sent after the brainstem as a corrective input for the motor command that is sent to the eye plant. So, what if we consider, for example, in this case, the feedback error learning controller? The problem is that, for example, there could be issue because the head and the eye rotate on different planes. So actually goes from the task error that you have in the eye that you can feel through the vestibular system. You cannot directly use that feedback in order to guide cerebellum for this. So what you need in such a case is an inverse function that is learned also by the cerebellum, which is actually a change in the kinematics, because you have an input coming from the vestibular system which accounts from the rotation of the head, but you need to move the eye in a different way because they are rotating on a different plane. The same happened also to the other path if we consider the recurrent pathway, but in this case you don't need for example, the kinematic inversion like you had in the previous case. And there have been studies which showed, for example, that for what concerning the feedback error learning, it is more robust against changing in the kinematics. Instead, the recurrent pathway is more robust in case of changing in the dynamics of the plant. So, how we use the cerebellum and where is the learning happening? We have the error that is computed, as I said, depending on which kind of architecture we use. If we use the feedback error learning or the recurrent architecture, and we have these updates of the weights in the Purkinje PFPC that is actually depending on the error that we have, for example, on the motion of the eye compared to the head, and the output of the granular layer. So we try to make a complete model, starting from this, from the study that we made on the VOR, that could account not only from the motion of the eye, but also for the motion of the head, because it has been studied that also the head is used during, for example, locomotion task, when you want to stabilize the image on the fovea. So it's not only the action of the eye, but there is also an head movement which is called the vestibulocholic reflex, which acts in order to stabilize the head in space. And as you see here, the cat is very good, it stabilized and reject the disturbance that come from the body. The difference between a human and, for example, animal like cats or chicken is that humans are not able to reject the translation that you have during the locomotion. This is due to the mechanics of our neck. So the neck can only rotate, so we can, in a way, reject the disturbances that are due to the rotation, and in order to account also for and reject the translation that happens when, for example, you are in the walking task, what happens is that the head rotates in the opposite direction compared to the translation that you have. So you have this, both this, what is called the linear vestibulo-colic reflex and angular vestibulo-colic reflex, according to if you are counteracting for the angular position, or if you want to stabilize with respect to the translation that the head is subject to. We integrate this vestibulo-colic reflex with the vestibular-ocular reflex that I showed you before in order to have a complete model of gait stabilization. And we also integrate another reflex of the eye, which is called the optokinetic reflex. This kind of reflex compensates for drift in the retinal image that happens due to external motion. This eye movement is very common, for example, when you are on the train and you look out of the train, you see different objects that move, and you try to stabilize the image by using this kind of reflex. So we made this whole controller for our robot. What we have done in order to test it was to move the trunk of the robot. In this case, we give, for example, a sinusoidal motion along the 3 axes of the trunk, and we use the neck and the eyes in order to stabilize the image in space. At the same time, we have also a target which is moving, and we apply the stabilization for neck and eye, and using also the optokinetic reflex, we could follow the target movement with the eyes. Here you see when all 3 are active, this is in red, should be visible, is the disturbance applied. And before was also possible to see that the error when we track the object moving on the screen was reduced when all the 3 reflexes were active. We did this also not only in simulation inside the neurorobotic platform, but we also implement this on a real robot. Here, in order to simulate the disturbance, we use this disturbing platform, you see, where we put the head and we control that platform, giving, for example, kind of sinusoidal motion or changing the frequency over time. And we did test activating only the VOR or activating VOR and the other reflexes that we implemented also in the simulated experiment. Another eye movement that we studied that where also the cerebellum has an important role is the smooth pursuit. The smooth pursuit is used when you want to follow a moving target. Differently from Saccade, this kind of movement is quite slow. It's called smooth pursuit. For this reason, it has not high velocity, but it is needed because when you want to follow a target, you want also to continue perceiving the environment. If the eye moves too fast, you have a blurry perception of what is going on in the environment. So this is used, as I said, to follow a moving target, and the goal of the smooth pursuit is to align the velocity of the eye with the velocity of the target in order to have 0 delay. Of course, in order to achieve this, you need a prediction of the target motion in space, which is done by the cortex, the medial-superior temporal area, but you need also an internal model of the eye in order to precisely generate a velocity that is the same as that one that is predicted by the cortex. So in order to implement this smooth pursuit, actually, we started from a simple controller where we have the visual processing, so we compute the position of the target on the retina, and we have this inverse dynamic controller, which actually is basically the activity of the cerebellum, which can control the eye plant and try to follow a target. Of course, the previous path had no prediction of the target, so this means that we try to follow a target, but we are constantly late because without an estimation of the next position of the target, we cannot be able to align the velocity of the eye with the velocity of the target. And for this reason, we needed a predictor like try to emulate the activity of the cortex that can produce the next position of the target that is needed to align the two velocity of the eye and target. If we have a look in detail, we start from the camera image that is processed in order to get the information about the target, and we compute an error information from this. Then we have the predictor, which is a very simple model, a very simple linear neural model. Actually, it has a tap delay input with all the position of the target, and the model of the neuron gives us output the next position in time of the target. Of course, the prediction in time is depending also on the dynamic of the eye itself. This means that if the eye, and we consider that the delay in the sensory system is, for example, 100 milliseconds, this means that our predictor should estimate the position of the target 100 milliseconds ahead. So what we do is using a simple decorrelation learning rule in order to make the predictor generating the next position of the target in order to be aligned. And here, for example, we show what happens when we have the input signal and the predicted one in space. The blue one is the input and the red one is the predicted signal. And we used also a method that allows to shift from a predictive smooth pursuit eye movement to a feedback one, because we should also consider that sometimes the motion of target cannot be predicted. If you consider, for example, that you want to follow a fly which is moving in space, its motion is not always predictable. So in that case, what happens is that we shift from a predictive smooth pursuit eye movement to a feedback one. And actually, we provide a kind of likelihood for what concerns the prediction of the target. This means that this value alpha says to us, okay, when it is close to 1, means that our prediction of the target is good, so we can rely on the prediction, and we can use that one in order to improve the performance of the smooth pursuit, because we are sure that the target is predictable. But when it, for example, goes again to 0 here, when the target becomes not predictable, as this, if you see the red signal after the 6 seconds, the motion becomes very kind of random. In this case, we should move back to the feedback control signal. And this is what we have implemented. For example, for the inverse dynamic controller, in this case, we used a simple robotic method, which is called an adaptive backstepping. And we used this inside our control loop. We did our experiment in simulation. Actually, in this case, we had a shift of the target motion. We changed, for example, the amplitude and the frequency of the movement, and you see here, when there is this change in the dynamics of the target motion around in around second 30, what happens is that our alpha value, which gives us the idea on how good is the prediction, goes to 0, because of course when there is a change in the dynamics means that our predictive model is not working anymore, and we shift to a feedback controller. But after a while, when again the target becomes predictable, we are able to reuse the prediction that are made by our neural model and align the target velocity and die velocity. This task actually also shows more or less the same thing, but instead of changing amplitude and frequency of the target motion, we go from a predictable one to another one which is not predictable. And of course, this has also an impact on the alpha that we use in order to estimate the predictability of the target. This is a video of example. We start with a motion, a predictive motion of the robot, of the target, like a pendulum motion. And you see in red, I hope it is visible here, the alpha value is increasing because the robot is going ahead more sure about the dynamics. But when we, for example, we disturb the target, the alpha value goes to 0, and we rely on a pure feedback controller in order to continue tracking the target also when it is not predictable. We used also Cerebellar model for other tasks, not only for eye movement. I showed you that it was possible to use Cerebellar model for the Saccades, for the VOR, and other reflexes like the vestibular colic reflex and the optokinetic one. And in the end, we show also how we used the Cerebellar model for the smooth pursuit. But we had also used and integrated for in a locomotion task. In particular, in this study, we want to make a quadruped robot walking, and we used a CPG algorithm which was based on a genetic algorithm. Actually, we use a genetic algorithm in order to generate the input to the motors, for the trajectory to the motors, and we use a cerebellum in order to adjust when the robot was walking the trajectory to make the walking more stable. As I said, we use nonlinear oscillator as a CPG, and relying on the CMAS, which is this evolution-based optimization strategy, we define genotype, phenotype, and fitness function, where as a fitness function we use the distance covered in 1 minute of simulation in order to understand what were the best locomotion patterns that we could have on the robot. We did two kinds of experiments. The first one we called adaptation in EVO. This means that we included the adaptive model that is represented by the cerebellum inside the evolution when we perform actually the evolution. In the other case, we first perform the evolution, and then after we get the best locomotion pattern, we apply the cerebellum in order to stabilize the locomotion. We compared these two strategies in order to understand what is more convenient, if it's more useful to have the cerebellum when we perform the evolution, or if it's enough to introduce the adaptive capability of the cerebellum when after the evolution is done and we do the test in, for example, in different environments. And what we found out is, for example, the adaptation in the evolution means that using the cerebellum also, the cerebellum algorithm also during the evolutionary part, it's more convenient because we have less error for what, if we consider here the top green graph show, for example, that we have less error for what concerns the joint considering the single joint of the legs. And we have also joint trajectories, which are the third graph in the matrix, that are more close to the reference, that is the red one. We also, considering the center of mass of the robot, which is the last row of the graph, we see that it is more restrained when we have the adaptation in the evolutionary part instead than after. One of the last work that we have done for using a cerebellum algorithm, we actually used the same one that we implemented for the Saccade task, but we used it in a completely different task, which is actually not really close to neuroscience. We decided to control a soft robotic arm, and we want to see if using an adaptive algorithm like the cerebellum, we are able to achieve better performance in the control of a soft arm. So what we have done actually is to use an approximated inverse kinematics model, which was implemented as a feedforward neural network, which actually was able to learn the relation between the task space and the input to our soft arm. And we introduced a cerebellar model, which gives change in the trajectories and see if this could make the soft robotic arm being more accurate when we try to reach a target in space. And what we found out actually is that in all the conditions that we had in our experiment, what we call CerebNet, which is the net where we included the cerebellum, compared to the others where we only have the inverse kinematics, we see that the error is smaller in all the case, in the normal condition, but also, for example, when we jam one of the actuators on the robot, the capability of the CerebNet to adapt, also when there are failure, in this we try to simulate the failure in, for example, in the robotic arm, in the top actuator or the bottom, or even when we completely remove one of the modules of the robot. You see the robot is composed of 3 modules. What we found out is that the CerebNet is has performance which are way better than the others. These are the work that actually we have done in the last years related to the cerebellum. As I said, we are now moving to this implementation of a spiking model for the Saccade, thanks to the help of Egidio and his group. And we think that these new models can bring new evidences and can help in understanding what happens in the experiment, for example, that I showed you for what concerns the Saccade. The goal that we have now for those experiments is try to replicate the activity of the Purkinje cell that we were able to replicate with the rate model, but we are sure that if we can do the same with the spiking model, it will be more valuable, and we can for sure also improve the performance that we had before. Okay, let me just thank my group and also all the partners of the robotic platform that work with us for developing, for example, the simulator I showed you before. Thank you.
So thank you, Gigi. I think it was a very nice presentation about the way the robots and the closed-loop controllers can be used to understand the cerebellum on one hand and to make functional artifacts on the other. Are there any questions? Take the microphone.
At some point, Egidio, you say that you, as error, you provide just a sort of direction of the error. So in the Saccade control, you say just if it's on one direction, on the other one. It's not completely clear to me then how you, I mean, the error can be in one direction, but a different amplitude. Yeah. So how you how then the modulation is tuned?
Yeah, I mean, this is possible because we use what is called distal learning in the algorithm that we implemented, and it's the way actually using this method it is possible not to rely on the magnitude of the error, but to use just the distance, and over trial it is possible to reach the goal even without knowing the magnitude of the error.
Yeah.
Okay, we are ready for the next session. Now we are in the session of applications and system integration, and the first presentation is from Alberto Antonietti on the modular control system, cerebellar connectivity and plasticity in PyNest.
Good morning, everyone. So in this talk, I will cover two aspects. I will start with the plasticities that we have embedded into the NEST model, the spiking neural network model of the cerebellum, and then I will show the connectivity and the modular structure that we have built and that we are going to test. So the integration of the two different aspects, the two different capabilities that the cerebellum is able to show. So as Egidio Falotico said in his talk previously, the two feedback and feedforward and inverse model that the cerebellum is able to do. But I will start with the general structure of motor control. So motor control has this modular structure and a hierarchical structure, and we have the different brain areas that cooperate and that are connected together to generate motor learning, motor control, all the stuff that we have seen in the previous presentation. And so now I want to focus on the cerebellum, and I would like to let's say show the network, the model that we have been developing in NEST, and we have the first input on the MOSI fibers that then goes through passing through the glomeruli to the granule cells, the vast amount of granule cells that we have in our cerebellum, and then we have this first loop in the granular layer, where we also have the Golgi cell inhibitory neurons that are excited by both granule cells through parallel fibers and ascending axons, and directly by the MOSI fibers. And then those inhibitory neurons, they inhibit themselves through gap junctions and back to the granular cells. So we have this first loop in the input stage of the cerebellum. Then the parallel fibers goes through the Purkinje cells, but also they go up to in the molecular layer to the molecular layer interneurons, basket cells, and stellate cells, which inhibit themselves and the Purkinje cell. Purkinje cell are then integrating information coming from the granular cells and from this second loop from the molecular layer interneurons, Then we have the second input from the inferior olive neurons, and this input is driving the plasticity through the clumping fibers at parallel fibers between granular cells and Purkinje cells and molecular layer interneurons. And then we have the last part of the cerebellum, which are the deep cerebellar nuclei, which receives excitatory input from the MOSI fibers, and they are inhibited by the Purkinje cell. And they have also an exchange of information of connections with the inferior olive neurons. And then we can read out from the cerebellum through the activity of the deep cerebellar nuclei. So this is the network, the model that we are using, and we are obtaining information about all these neural population from physiological data. Then this model has, let's say, a static behavior, so some firing rates of the different neurons, but then it's able, and the cerebellum is the most plastic structure of the brain, so we needed to have plasticity. And plasticity not only at a single site, let's say the most studied one, which is between parallel fiber and Purkinje cell, but we wanted a distributed plasticity. So we have identified, well, in the cerebellum there are more than 15 known forms of plasticity, some experimentally measured, something with direct or indirect measurements. And we have included some in a distributed fashion way. The first one is, we can call them the cortical plasticities because they are limited to the cortical cerebellar cortex. So where Purkinje cell, granular cells, and molecular layer interneurons are. And the first one is between granular cells and Purkinje cells, so parallel fiber Purkinje cell plasticity. And the second cortical plasticity is between parallel fibers and molecular layer interneurons. And then we have also nuclear plasticities, nuclear because they involve the deep cerebellar nuclei. The first one is between the MOSI fiber and the DCN, so shaping the excitation of deep cerebellar nuclei. And the second one is between Purkinje cell and DCN, shaping the inhibition coming from the Purkinje cell to the deep cerebellar nuclei. And then we have a further plasticity at the very input stage of the cerebellum between MOSI fibers and granular cells. So we have developed those plasticities, and each one has a specific behavior, and we try to model it with following the biological data. So we start with the cortical plasticities. We have modeled all those as long-term plasticities, so with long-term potentiation, LTP, or depression, LTD. And the learning rules that each plasticity follow is a mathematical equation that tries to mimic the biological behavior. So for example, for the parallel fiber Purkinje cell, we have a change in the amount of the long-term depression that depends on the spike timings of the neurons involved in this plasticity. So we have three populations because this is an heterosynaptic plasticity. So we have granular cells and Purkinje cell, and then we have a third population, a modulator, which are the clambin fibers coming from the inferior olive. So we have LTD when we have an error in the system, let's say, and the error is caused by the clambin fibers. So we have a change in the shape, in the weights of parallel fiber Purkinje cell synapses that depends on the timing, the difference between the inferior olive spike and the granular cell spike. So we can see that there is this peak of LTD, of depression, around 100 milliseconds. So the parallel fibers which were active before in this window, before the arrival of the clambin fiber spike, they change their weight, they decrease their weight according to this rule. While we have an LTP every time a spike goes through the parallel fiber. For the molecular interneuron, the mechanism is similar, so it's again a time difference. All of those are variations of STDP, spike time dependent plasticities. And in this case, we have a change not of the LTD, but of the LTP. And this change, so the amount of increase in the weight, depends again on the timing. And in this case, the peak is around 50 milliseconds before the arrival of the clambing fiber spike, or the inferior olive spike. And we have reproduced those kernels in NEST. Going through to the nuclear plasticities, the MOSI fiber DCN, in this case, we also have heterosynaptic plasticity because we have MOSI fibers, DCN, and the third population, a modulator, which are the Purkinje cells. So the Purkinje cell spike, which is where we have centered the kernel, when the Purkinje cell spike happens, then we have a decrease of the strength of the MOSI fiber to DCN synapses for the MOSI fibers that had a spike very close. So you can see that this window is -10, +10 milliseconds. So if we have a very close spiking of MOSI fibers and Purkinje cell, then we have a depression of this plasticity, while also in this case we have a potentiation every time a MOSI fiber emits a spike. Then the Purkinje cell DCN plasticity, this is the, let's say, the standard STDP plasticity. So we see that we have both on the same kernel, the LTP and the LTD, and the LTP or LTD depends on the causal or anti-causal mechanism that we have. So if we have before the Purkinje cell and then the DCN spikes, so the presynaptic and then the postsynaptic firing, or vice versa, we have LTP or LTD. And then we have a more complex plasticity, let's say, at the input stage of the cerebellum between MOSI fiber and granule cells. So we have experimental data coming from Egidio's lab that where they have measured the changes in so the LTP and LTD according to the differences of spike timing, so the STDP, and we can see that we obtain this rule that is similar to the last one that we have seen, the PC-DCN plasticity. But they also observed that the behavior changes according to the frequency, of the inputs. So it's not the same. It's not only the spike timing, that matters, but also the frequency of the input. And this is coherent with all the findings about the oscillations that are moving through the cortex to the cerebellum and inside the cerebellum itself. So we have two mechanisms that we wanted to include in our plasticity. So we needed to model both the STDP dependency and the frequency dependency. So we have the experimental data, and we needed to transform those data into equations. And so in this case, we have done a fitting. So we extracted the equations that fitted the experimental data for both the time dependency and the frequency dependency. Then the time dependency is, let's say, easy to implement. It's a standard STDP, but then we need to take into account also the frequency dependency. And for the frequency dependency, let's say the synapse has to do an additional work. So it has to compute the oscillation frequency of the input. So it has to compute first the instantaneous firing rate of the input. Then it has to understand if there are oscillations in that instantaneous input frequency. And then, according to the oscillation rate, shape or modulate the amount of LTP or LTD. So, starting with the first part, we needed to compute the firing rate, the instantaneous firing rate. So we have tested different approaches, and we found that a good solution is to having this function that increase every time we receive a spike. So if we have more spikes, the frequency is increasing, and then we have an exponential decrease. And we have those two parameters, step and tau, which according to the different parameters, we can have a different behavior. And then we optimize those parameters to have a function that can keep track of different frequencies. So we have tested different parameters. We wanted to be able to track the input frequencies of 5 up to 100 Hertz. So we have optimized those parameters to minimize errors, and then this is the result that we obtained. So we are able to have a good measurement of the instantaneous input firing rate. And then we need to compute the power spectrum of that signal and to compute the amount of the oscillation frequency. So we tested this plasticity with in different conditions, with bursts of spikes, with different oscillation frequency, so with different time intervals between the burst, from 1 to 10 Hertz. So we stayed in the theta band, more or less, the frequency band that experimentalists use in their experiment, and with different intra-burst frequencies, so the frequency within the single burst. And then we applied an FFT algorithm to efficiently compute the power spectrum to identify which were the frequency components of the input signal. And then we wanted to minimize the error between the computed peak frequency, oscillation frequency, and the one that we were applying. And then we needed to combine the STDP rule with the frequency dependency rule. So this is the behavior of the synapse. So you can see that those 2 points, which represent the delta t, positive or negative, between the firing, you can see that according to the input frequency, the shape of the kernel changes. And for example, with frequencies which are less than 2 Hertz, we just have LTD. Even if we have a positive delta t, we have only an LTD with very low frequencies. So that's what we have done. So this is how we tested the plasticity, and we wanted to see if adding an error, a random stimulation, or having a variability of the oscillation frequency, how robust was the algorithm. So we can see that with a high number of input spikes with a very high frequency, so we have very clean burst of spikes, the performances of the algorithm are ideal. So if we apply an oscillation frequency of 1 to 10 Hertz, we always obtain the correct, we identify the correct peak. If we use just one spike, so not even a burst, and very low frequencies, well, this is the same for all the first column, we obtain very bad performances, of course. In an intermediate situation, we obtain some correct values and some wrong ones. And then we tested the robustness, so we added a variability of the interburst interval, so a variability in the frequency, and with let's say a variability of 5% or 10%, still we obtain a reliable frequencies. Of course, with 25 or 50% of variability, then the error increases a lot. And if we add an additional noise, of course, the errors are even bigger. But if we have a limited amount of noise and a limited to 10%, up to 10% variability, then we obtain good performances. Then the next steps that we are going to develop is to include an additional feature. So this synapse was complex enough, it wasn't complex enough. We needed to add another behavior, so we needed to take into account also the some, let's say, molecular level mechanism. So for example, the impact of the nitric oxide, NO, which which enables or disables the plasticity. So this is a further mechanism that enters in the field. And this nitric oxide has a certain distribution in the space, and it's released when we have a postsynaptic activity in the granule cell. And it has also its own dynamic, temporal dynamics, which depends on how strong is the firing of the granule cell, and there is a relationship between the NO release and then the CGMP activation with a different dynamic. So it's a complicated mechanism, and we are working with Alessandra here, Alessandra Trapani, to add this mechanism to the already complicated, let's say, mossy fiber granule cell synapse. But we want to be as close to the biology as possible. So this is something that we need to take care of. So also here we have a dependency on the frequency. So brain is very complicated. So I conclude the first part on the different plasticities that we have, and I will be back to I'm going back to this initial slide about the modular control system. So we have seen the different players that cooperate to generate a behavior. And as we also seen before, it has been proposed that the cerebellum implements both the inverse model that transform an input instruction or a sensor information into a motor command that goes to the muscles, and a forward model that generates a prediction of the sensory state using the information about the commands that are sent to the muscles. And this modular approach is something that we want to implement with a realistic network as the ones that we have seen in the previous days. And we have been inspired by an experiment on human subjects done by the group of Honda in Japan. So they used this experimental setup where a subject needed to touch a screen, a point on the screen, but while wearing prismatic glasses. So what he was seeing on the screen wasn't really where he thought to go. So at the beginning, the subjects committed errors on the position where they touched, and then gradually, thanks to the adaptation gave by the cerebellum, the error gradually decreases in some tens of trials. And then they tried different experimental conditions in which they kind of switch off one of the two modules of the cerebellum, and they've seen different dynamics in the adaptation. So this is what we would like to do with a realistic model. So the system that we have designed, and this is a work that also Eduardo is doing with me, is to define also the other parts of this corticocerebellar loop. So for example, the motor planning that transform the intention to go in a certain place to spikes that are then sent to the motor cortex that generates the motor commands. These cortical commands goes to the robot motor system or to the muscular system, and then we have an action, the generation of the action, and then the sensory information recorded by our sensory system or by the robot sensors. And here we can do this, let's say, without the cerebellum, but without the cerebellum we cannot compensate for an error. The prism, the prism of the glasses, it's an error that we cannot know, and we cannot adapt if we don't have the two modules of the cerebellum. So we also receive the position, the real position that we wanted to reach, so we know that we are committing an error. So we have this first module of the cerebellum, the forward model, which receives the reference copy from the motor cortex, and it generates a prediction of the error that we are going to commit. And then the inverse model, which receives the information from the planner, so the intention, and it generates to the robot motor system the motor correction. So at the beginning, if we are doing the task correctly, these two modules of the cerebellum are silent, they are not needed. They are activated when we have an error, so when we have to compensate for this error. So when we also have the information of the proprioception coming from the joint, we don't have the visual feedback because during the movement the subject doesn't see the point that he had to touch. And then according to what we have done and what we were supposed to do, we can have an error that can be both sensory or motor, and then it used as a teaching signal to the two cerebellar modules. So the planner, which can be seen as the premotor cortex, transform a desire and intention to a spiking pattern, because we are working with the spiking neural networks, that is sent to the motor cortex and to the cerebellum. And this is a firing rate coding about which is the distance that we need to cover, let's say. And we have a linear dependency of the firing rate of the planner with the desired position that we want to reach. Then the motor cortex receives from the planner and also from the forward model of the cerebellum, so the sensory information, and it generates torques, it generates spikes to muscles, and that also goes as reference copy to the cerebellum. So having the spikes that enters, they encode the amplitude of the movement. So for the 4 joints of the arm, we have a different amplitude of the movement that we need to perform, and according to the firing rate, we have a different amplitude that we want to perform. Then if we derive 2 times, we obtain the accelerations that are needed to perform that angle movement. And these are transformed, decoded into spikes, encoded into spikes according to this, let's say, somatotopic conversion, where we have positive agonist and negative antagonist motor commands that goes to different muscle fibers that have a different strength. So we have a sort of weighted sum of the movement. And then the robot receives this information from the motor cortex and from the other module of the cerebellum, the inverse model, and it transforms those information into a movement. And to do so, we have this counting, this kind of integration with the gain, depending on the positive or negative, and how positive and how negative is that muscle fibers. And so we can reconstruct, let's say, the signal. We can see the reconstruction made by the system and the original signal that was the plan. And if we integrate, we can obtain the movement, the angular movement. Also here we have the obtaining one and the original one. Of course, we have some degradation of the signal due to the conversions between spikes and analog signals. And then it has to generate the error to the two parts of the cerebellum. So we have a sensory error, and it can be encoded, let's say, it's a linear error, so according to the sign, to the direction, if we have gone to the right or to the left of the target, a large or a small error activates different fibers. And while the motor error, let's say it has a shape because it has a sort of template, stereotypical template that is then modulated by the amount of error. So if we have a small or a large error, and if we have a positive or a negative error, we have to activate before the agonist and then the antagonist, or vice versa. So this is how we have developed the other parts, the other modules that are connected to the cerebellum, and the cerebellum, the two cerebellum models are the ones that we have seen in the previous days. So with the scaffold generation, biophysical inspiration from biology. So thank you. If you have any questions, I'm happy to take them.
Hi, thank you for your presentation. Could you go one slide back from here? I was wondering, the sensory and the motor error, are those the spiking rates of the neurons, and how do they then represent the error? I didn't get that part.
Okay, so these are, let's say, related to what I need to add with my cerebellum. So here we have the two modules. So the sensory error goes to the forward model that has to generate a sensory prediction. So from the planner, I receive the information, you have to go at the position A plus P. But then when I receive the feedback, I see that I didn't want to go there. I wanted to go to A. So I have this linear error, and this is, let's say, an error of my sensory system because I have the glasses. So I need to compensate for that error, and I have to change the signal that is the output of the planner, which is firing rate approach, and I have to increase it or decrease it according if I am at the right or at the left of the target. And this is why I have a firing rate modulation of positive or negative, let's say, inferior olive. While the motor error goes to the inverse model. And the inverse model has to generate a compensation on the motor part. So it has to behave, let's say, similarly to this shape. So we wanted an acceleration and then a deceleration of the movement. So we are moving our arm not in the right way. We need, for example, more acceleration, more deceleration, so a more wide movement. So we need to increase the acceleration and deceleration to reach the real ideal point. Or we have gone too far and we need to reduce, so we have to reduce the acceleration and deceleration in the same way. So that's why we have this kind of template-based and firing rate approach together.
So what you see in the plots are the differences, what should have been the motor command and the sensory information?
Exactly,
Okay.
yes.
Yeah.
Yes.
Very nice presentation. Can you go back to these slides? Right. So by the end of the learning, we expect the two pink input to be 0. So the cerebellum models are no more training. And then it's not fully clear to me what governs the compensation which has been learned. Did you move it back to the motor cortex or is implemented by the cerebellum inverse model?
No, all the changes are limited to the two modules of the cerebellum. We don't have a transfer of the learned correction back to the cortex. So all the learning happens in the two modules, and if we have reached a plateau, let's say, in the movement, so we maintain that correction, let's say, forever. If we have a change in the glasses, so if we shut down the glasses, so we have to go back to the previous situation, and then we have a change. Again, there will be errors, so there will be a new adaptation of the two modules to the new condition. But we don't have a transfer between the two models and the cortex.
So at the end of the learning, if you are in a stable situation, the output of the cerebellum forward model will be 0. You don't have any sensory error prediction. And the compensation is driven by the cerebellum inverse model. Is that true? I mean, we have to think about it.
Okay.
But,
probably not, in the sense that both models learns a part of the task. Let's say that we reach the ideal behavior with both the contribution. So we reach the optimal behavior because our sensory information going to the cortex is better, and partly because the correction that we are doing is helping. So in theory, we should we have to see what happens, because we haven't started yet the real optimization. So maybe there can be that at the plateau, then the information from one model switch off, let's say, and everything is done by the second one, or both keeps going. So that's a nice thing that we need to explore.
Anyway, it doesn't take into account the motor correction from cerebellum. So if the movement is correct since the sum of the two, anyway, just the motor cortex somehow is not enough. So the output, the sensory error prediction, is probably not 0 anyway. Even
yes.
Okay. So probably difference copy could be also the sum of the two. It depends. In this case, it's just the output of the motor cortex.
Yes.
Which is just one contribution.
To to which detail do you model the motor cortex? Do you have like a microcircuit model, or do you have multiple areas?
Okay, well, right now, the model that we have, the motor cortex, is just, let's say, equation-based. So it's not a spiking model. Of course, it will be very interesting to use, let's say, a biophysical model, a realistic model, as the one that we use for the cerebellum for the cortex. And this is for the next years, probably. But yeah, we will need a functional model of the motor cortex to use it in this kind of loops. But that's a plan,
right,
long-term plan.
Very nice integrative work. I was wondering how the planner is implemented. Maybe I just missed it.
Okay, I can go back. So the planner is kind of simple. So according to what we see, so the target position plus the deviation from the prism, we know where we want to go, and let's say the conversion is firing rate based. So if we want to go at 50 centimeters, it's a certain firing rate. If you want to go 60, it's a bit higher, and so on. So it's kind of a simple way to encode the amount of where we want to go. This is the simplification that we have done.
Can I just point out the last comment? The difference between sensory and motor error is implied by the needs given by the two modules in the cerebellum and the different kind of information that is needed by the modules to elaborate the error. So one may ask, is it what happens in physiology? Do we have two motor errors or not? So it is something that is needed by the model because we don't understand something of physiology, or it is something that exists. So the point is that it's probably something that exists because there are different pathways conveying errors, or I would say not maybe error at the beginning, but sensory information from the spinal cord to the brainstem to the cerebellum. There are multiple pathways, and these are well known to be differentiated. They do not carry the same signal. Nothing happens by chance in biology. There is no space, there is no energy, evolution eliminates all what is not needed. So what remains is needed. There are different pathways running from the spinal cord to brainstem to cerebellum, and one of these is done in a way that accumulates information coming from the motor system and the sensory system. They put them together. This is probably the place where the motor error is generated. So there is some place already in the spinal cord that generates the motor error. And for the sensory error, it's easier because it's just sensory. So probably there is a corresponding anatomical and physiological substrate to what Alberto is modeling. And then the other thing that I would like to point out is that the model he presented at the beginning of plasticity in the granular layer is very new. There has been no corresponding attempt to introduce plasticity in the granular layer so far. And I think this is the last point, or I wouldn't say the end point, but the last point of a long series of observations started at the end of the 90s when this plasticity was discovered. And nobody has been able ever to understand what this plasticity is doing to the circuits of the cerebellum. Consider that this is the most plastic site of the brain. It is the most extended site of the brain, and nobody knows what this plasticity is doing. So I think this is a very important point, a very critical point. So if the models done in this way will be able to tell us something about the role of this plasticity, that would be great. So, well, that's just to point out the relevance in the physiological context. Okay, if there are no other questions, issues, or comments, we move to the next from Francisco. Is Francisco or is Alessandra? Is Alessandra? Because the names on the program are Francisco and then Alessandra, but that is Alessandra and then Francisco, okay.
So, good morning. Thank you, Egidio, for inviting me in presenting this contribution. The contribution I'm presenting is related to the activities of CerebNest, which is one of the partnering projects of the HBP. And it has been conducted in strict collaboration with the University of Pavia and EPFL groups. So I want to start from one of the most important slides I've seen in Egidio's presentation yesterday. He didn't send me the right one, so I took my picture. And
sorry.
So Egidio was indeed pointing out two key points, which are, in my opinion, of paramount importance in the whole story we are telling. First, model construction data, which are used to construct, to build the model, must be different from the data used to validate it. And this is a very key point. And the second one is, when we talk about brain simulation, data as well as models need to face a multi-scale approach, and they can talk each other at different levels of this multi-scale. So we can have molecular data, neuronal level data recordings, microcircuits level data, and behavioral data. And models can talk to data at these different stages of the scales. And so what we indeed have been working on is about giving validations and, do you see my pointing? Yes, okay. Embedding computational models into simulations, which can be done in silico or with real robots, so to reproduce a behavior. So neurorobotic represents one of the possible link between data and models. And what is the hallmark of neurorobots? It's that the robotic behavior mimics the features of brain-controlled behavior. And these features are actually some of the most typical capability of the animal and the humans, like timing, smoothness, generalization, learning. And we have seen a lot of examples in Egidio Falotico presentation this morning, as well as in Alberto framework presentation. So the link between model and data can be in two ways. So we can read this loop, we can read this loop starting from the behavior and going counterclockwise to the models. So the red direction starts from setting the features of the expected behaviors as seen from, for example, biomechanical data or behavioral gait analysis, eye tracking data, and then develop a controller of the robot which assures the achieving of the behavior, and then associate the parameters and the construction of the controller to the brain structures which are supposed to control the neurophysiological behavior. The other way around, we can read this same blocks by starting from cell recording and microcircuits recording, building the model, embodying the model into simulations or robots, and then analyze the behavior as an emerging properties. And in this way, we close the loop against on data by ending with the behavior. So we can start from behavior, build the controller, and look at the controller, comparing the controller with the cell recordings. Or we can start from cell recordings, build the controller, and see the behavior as the validating framework. So these two approaches are not one superior to the other, are just different. And they both use different data for constructing the model and for validating it. So the loop we are using is the one starting from cell recording and going to the behavior as an emerging property. So our building blocks starts from the model, and the model is done by the architecture of the networks, the single neuron activation functions, the connectivity between the different populations of the neuron, and then the plasticity rules among the different connections. And in this way, what Alberto was explaining in the talk before me is exactly what I mean as this is the building block of a model. So we start from the model, which has been inspired by cell recording, neurophysiological data, and informations coming from experiments from the single neuron populations and microcircuits. Then, the second very important building block is about protocols. So we need to embody the model into a target features, a target behavioral features. And to do this, we need to have clear ideas on what the model is expected to control. And in this framework, the cerebellum is one of the most important test bench for neurorobotics because we have some protocols, some behavior which are clearly linked to the cerebellum properties. And these are tasks for association of timing, learning, and prediction. And we can use what has been studied for years by clinical neurophysiologists in order to study cerebellar pathologies, and we can translate the tests, the protocols that clinicians used to test people for cerebellar disease and translate this protocol into the testing protocol of our controllers. If it works, the cerebellum is working. If they are not doing the protocol in a proper way, then their cerebellum is pathological, is not correctly working. So, three main stories about protocol are in the cerebellum community. One is about eye-blinking classical conditioning, so a pure timing association. Then we have seen a lot of examples about vestibulocular reflexes this morning. And the third area is about limb movement, arm reaching in perturbed conditions. So, for example, in force fields. And so these protocols needs to be converted into input and output to the cerebellar model, to the spiking cerebellar models. Let's go through a couple of examples. In the case of Blinken classical conditioning, which is the most used protocol, we have a conditioned stimulus, such as a tone, and then an unconditioned stimulus, such as an air puff, which forces the eyelid to close. After a certain number of repetitions, the association between the tone and the air puff is learned by our cerebellum, and so the eyelid closure, so the conditioned response, occurs just before the air puff. So the cerebellum learns the timing association and uses it to predict the conditioned response. So in the model, we need the tone to enter the mossy fiber, while the air path is the unconditioned stimulus going to the inferior olive and then conveyed through the climbing fibers to the Purkinje cells. The output, which activates the eyelid closure, is provided by the DCN. So here we can see that this protocol can be converted into a protocol of the now robots. So the conditioned stimulus is the tone, and the unconditioned stimulus is the laser, which is hurting now. And what now needs to learn is the association between the two in order to give a conditioned response, which is to move the shield and protect himself from the laser. And you will see that after a few trials, so exactly as the cerebellum, we need trials and repetition to learn. Now it has learned to activate the shielding action. And then learning the conditioned response, the cerebellum also is in charge of learning the extinction of a conditioned response. So when the two stimuli are no more associated, we need to abandon the behavior. And so as well, the robot, here the laser is no more present and the action of protecting is no more required. So the robot needs to abandon the learned behavior. And all this is done by the spiking neural networks. So something like what you have seen in NEST, in all the previous presentation of yesterday. Another protocol is about reaching in force field. So here, the protocol responds to a very cerebellum-focused feature. So if you have to take a luggage, you always have a good estimate of the weight you are going to carry. And this estimate comes from your experience. And so now here has to follow a track, and at a certain point, an extra load is put on his hand. And so in order to follow the track, he needs to learn to compensate for the perturbation given by the extra load. And so here you can see that at the beginning now learns to follow the track in a very baseline situation. So you can see the desired trajectory in blue and then the one done by the robot in red. After a few trials, there is the load, the external load put on the end effector of the robot. And so here you have 10 kilograms put on the hand of the now, and now starts to going below the trajectory and needs to learn to compensate. Of course, here we haven't put any feedback controller. So the cerebellum is the only area is the only controller which allows for the compensation to happen. But the cerebellum needs repetition and learning to do this. And after a few trials, you will see that the error is reduced. There is not a full compensation because there are some joints of the now which are not controlled by the cerebellum for keeping real-time control. And the problem about real-time will be then talked and deeply discussed by Francisco. So the point is, we need to embed more and more increasing complexity into the models. And in order to be able to really validate them against behavior. To do this, we have seen that we need more detailed point neurons, we need physical simulator and robot controller, and actually HBP is giving us a lot of infrastructures where this kind of testing can really happen. So I will just run through some tests that we are now doing on the neurorobotic platform. The neurorobotic platform of HBP is giving us the possibility to control virtual robots, which means that still we have a body and we have a behavior, but we are losing the constraints about real time because we are playing with virtual robots. But the main advantage is that we can embed very complex models developed in Nest. For example, here we have the eye blinking classical conditioning, so the same as the now shield, the now using the shield in the neurorobotic platform, where we have embedded the spiking neural network of the cerebellum. And so the car has to learn that it's like a parking sensor. So the red light means stop, otherwise you will crash. After a few, so starting from the green light, the red light indicates the unconditioned response. And after a few trials, the car learns to stop at the red lights without crashing against the wall, so to avoid the crash. We are also working on a new protocol about mouse whisking, which is about learning, having a go and no-go trials depending on the position of a pole, which is read by the whisking. And so we have put on the neurorobotic platform, there is a mouse, but it didn't have any whisk, so we put the whisking, and we are now adding the trigeminal ganglion and the facial nuclei. And so as you have the virtual robot, you need to build the brain structure for controlling all the parts. But I'm now concluding, and I want to go back to the very rationale of the whole story. So the eventual goal of neuroscience studies is to know more about the brain, with the final objectives to develop better solutions for brain pathologies. So this is the very final goal. And I think it's important to try to understand how can brain models and simulation and neurorobotics contribute to this final framework. And so I think first, robotic embodiment allows to validate models built on neurophysiological cell recording data on target behavior. And as models are validated, we can apply specific diseases to the model. And so we can mimic the pathological condition. And so we will have pathological models, and we can see the simulated emerging behavior, which can be validated against misbehavior of patients. But once we will have this kind of parallelism between the patient and the model, this will open a lot of possibility, because in the model we can look inside and we can see all the mechanisms. We can play with the model, trying to activate compensating mechanism inside the model and derive from this gaming in silico or with robots new direction for therapy for patients. And since my other day I design technologies for rehabilitation, I think we need to keep the final goal in our image. So thank you very much. And one example of these neuropathological robots is a very nice work by Alice in 2018, which is 17 actually, and which is a very nice test of this approach. So thank you very much. If you have questions, please.
Thank you very much, Alessandra, for completing the picture of the work. Are there any questions? Okay, probably all the questions have been asked before. And so thank you very much again. And now Francisco Naveros, always on the Cerebellar networks in closed-loop controllers and real robots.
I'm going to present a couple of experiments. In this case, the first one is a POR experiment, in which we are more interested in understand the computational mechanisms used by the cerebellum to perform this behavioral task that we emulate with the robot. And the second experiment is a robotic arm manipulation task, in which we are more interested in the control system itself and how it is able to control compliant robot with even better result than traditional position control method. Well, this kind of experiment can be easily implemented using simulated robot, for instance with the neurorobotics platform. But when you try to implement this experiment with real robot, you have to deal with a lot of problems. For instance, non-linearities, noisy sensor, noisy actuator, and the most important, real-time restriction requirement. So in our experiment, we use EdLUT. EdLUT is a spiking neural network simulator, mainly developed to implement this kind of closed-loop neurorobotic experiment with real robots in real time. EdLUT can implement many different point neuron models, like interactive 5, ADEC, HH, distributed plasticity. This simulator implements parallel CPU and GPU parallelization mechanism. And also we use a hybrid event-driven and time-driven simulation method for the neural model and the learning route. And finally, EdLUT integrates a real-time supervisor that ensures that the simulation is going to run in real time. It's able to speed up or slow down the simulation to fit this real-time requirement. Well, as Gidio told yesterday, this kind of cerebral closed-loop controller always include or combine a spiky module, for instance, the cerebellum, but also a lot of analog module that emulate different body regions, such as associative cortex, motor cortex, brainstem, body sensor, and so on. Well, in this analog module can be internally implemented on ELUT using the ELUT robot interface integrated ELUT, or can be externally implemented in a ROS interface, and in this case, ELUT just simulates the neural network, the cerebellum. Well, this is the first demo that I'm going to show. This is the VUR experiment with the neurorobotic platform. So the VUR, the cerebellum compensates the hand movement. Here is the control, the closed loop control scheme implemented, the spiking cerebral model and the transfer function with all the analogical function. And we can see how at the initial learning stage, the cerebellum is not able to compensate any movement, so we can see how the eyes are moving with the head. And we can see also the activity of climbing fiber, vestibular nuclei, Purkinje cell. And then, after the learning, at the final of the learning state, the cerebellum is able to compensate the movement and stabilize the image. Okay. We also implemented this experiment with ELUT and with our real ICAP. This is the control scheme that we implemented in ELUT. In this case, everything is implemented inside ELUT with the robot interface. And this is the experiment, the VUR experiment. In this case, the ICAP robot is controlled using velocity command. So it's quite more easier than torque control that we will use in the second experiment. So the cerebellum controls the velocity of the eye. Of course, it is able to learn different amplitude and different velocities, both in horizontal plane, but also in vertical direction. So here we have a representation of the optical flow, the movement of the eye, the error. Here we can see how the image is stabilized. Okay, with this experiment, we try to understand the complex spike in Purkinje cell, the effect that this effect had in the behavior of the experiment. The second experiment that I want to show you is a robotic arm manipulation experiment. In this case, we use a compliant robot. That is a robot that integrates elastic component inside of his structure. And this kind of compliant robot are really hard to be operated using traditional position control method. Actually, all Cerebellar implementation achieve better performance result than this traditional position control. So in this case, we are more interested in the control system itself for implement robotic application for the future. The control scheme implemented is this one. We have the trajectory generator that generates analogical signal, the desired position velocity signal, and also the Buster robot that generates the actual position and velocity signal. We transfer all this analog signal to a spike and processing the cerebellum. The cerebellum generates the output activity that we transfer to torque motor command that we send to the robot and minimize the error right to the cerebellum through the climbing fiber. So in this case, we implement just a learning rule in parallel fiber to Purkinje cell due to computational restriction. In the VUR experiment, we have a plasticity mechanism in parallel fiber, but also in MOSI fiber to deep cerebral nuclei. Well, this is the experiment with the neurorobotic platform. This is the neural network. He has 6 different microcomplexes in the cerebellum. And here we can see the neural activity of the 6 microsomes of Purkinje cell, deep cerebral nuclei, and clambering fiber. In this case, the task is a circular trajectory with the N effector. So the Cerebellar model must control 6 joints of the robot to perform this task. Here we can see both the desired position of each joint, position trajectory, but actually the actual position. So at the initial learning state, we can see how the cerebellum is adapting the neural activity and modify the trajectory, and then at the final learning state, we can see how both current and desired trajectory perfectly match, and the robot is performing the trajectory.
Sorry, Francisco, can I ask you, what is the cost function here? So how does the robot know how to optimize the movement of the different joints?
In this case, we use the mismatch between the desire and the current error. We don't have an inverse kinematic model. Just the cerebellum performs everything. It's a simplification also because we have limitation in the computation. We would like to include an inverse kinematic model to control the movement, and the cerebellum just synchronize the movement and learn different objects. But in this case, the cerebellum performs everything.
Okay.
It's a simplification. Okay. And here is the cerebral model that we have implemented in ELU to perform the same experiment, but with the real robot. We are able to simulate 62,000 neurons and more than 36 million of synapses, most of them with plasticity mechanics, in real time. And here is the experiment. In this case, the task is a egg-like trajectory with the N effector. The ROS interface allow us to plot all this information online. So this is the activity of deep cerebral nuclei. This is the motor command, the torque motor command. Here in blue, we have the desired trajectory, and in red, the actual trajectory in real time. And here, we see the error, the mean absolute error. The red one is the error with our cerebral model. And this blue, this dash blue line is the traditional position control method. So we can see how, in this case, the cerebellar model achieves better performance result than that position control, that native inbuilt control. So we can use this technology to implement robotic tasks more efficiently in this kind of compliant robot. Okay, thank you for your attention. Any question?
No, maybe. No, Claudia.
You showed the two experiments on the simulated robot, so VOR, ICAB, VOR, and circular tracking. In that case, you use directly PyNest. You don't do any ADLU translation because you don't need any real time.
We implement both experiments, both in the neurorobotic experiment.
Yeah, in the neurorobotic.
But in ELU, we use ELU always.
For the real-time robot, yes. In the other case, directly PyNest network. Okay.
Yeah, one question, Egidio. So I also noticed that this is one of the rare occasions in which there are two Egidio in the same room, because this is a very rare name. And okay.
Yeah, the implementation are very nice, and also the model is impressive. I have the question is a bit broad and relates to both the implementation. I saw that actually, I was wondering what is the generalization capability of the model, in the sense that the feeling, at least from the experiment that I saw, seems that you use for learning and testing the same input. Means that, for example, for the VOR, you learn on a specific amplitude and frequency disturbance, and you test on the same one. I'm wondering if this cerebellum has a generalization capability, in the sense that it can really learn the inverse and model, forward model. If you change the trajectory in input, is still working, or what happens? And the same could be applied for the control that you use for the arm. I mean, if you change the trajectory, it's not anymore that one, is still working or needs again to learn?
In the VOR experiment, we have tested 3 different amplitudes, and it's always the same Cerebellar model. And in the robotic arm experiment, we have implemented 3 different tasks: a circular trajectory, an egg-like trajectory, and target reaching trajectory with the same neural network. So the model is able to...
You don't need to learn again. I mean, after the learning is finished, you don't need to learn again when you change the trajectory, or you need again another...
You can learn other trajectory. It is not destructive. You can learn simultaneously a circular trajectory and an egg-like trajectory.
No, I get this, but when you change trajectories, you get an error? There is needs still to improve, or when you finished your learning, the controller is ready, whatever trajectory you have and whatever disturbance you have for the VOR?
You can implement whatever trajectory. You have a desired trajectory, and you compare the desired trajectory with the current trajectory and compute the error. And we use this error to codify in climbing fiber. And we also inject by MOSI fiber the desired trajectory and the current trajectory. So we generate in granular cell a sparse codification of the space of all the joint, and this sparse codification is correlated with the error coming from the climbing fiber. So it's able to learn whatever trajectory, whatever task.
Okay, thank you.
Alessandra?
I just want to acknowledge that we as well, when we talk with the real robots, are using EDLUT. So it's Granada development. And then I would like to comment the importance of the last part that you have shown, which is about how this neuro-robotic approach can open to new robotic design. If you want to give a take-home message about this point, I think it's very important.
Okay. Well, this kind of technology can be used for this collaborative robot. This collaborative robot have a lot of problems to be controlled using this traditional control system, and using this new technology, this new approach, using biologically inspired mechanism can be an alternative solution to control this kind of robot. Additionally, this robot can interact with humans because they have low power actuator, and it is possible to interact with them without any risk for humans. I think this is the kind of technology that we will have in our house in maybe 30 years ago or something like that. So we must research in this technology and this kind of control system.
So I haven't, oh, please. No, I stay last.
Thank you for your presentation. I had a similar question to what has already been asked. Like, when you have a new trajectory, because you say it can learn any trajectory, but eventually the 8-like trajectory it was able to perform it after like 400 trials. If you have then a different trajectory, does it have to start from scratch and again have 400 trials to perform that trajectory?
Yes, but when you return to the first trajectory, you have not destroyed the learning. So you can learn several trajectories at the same time. And learn one trajectory doesn't destroy the learning before.
Okay.
Because each trajectory it is codified in different granular cells and in different parallel fiber to Purkinje cell synapses.
Okay, thank you.
Oh, yeah.
It's a kind of continuation to this question. So if say learning a new trajectory destroys the learning from the previous one, or let's say it's not useful for the previous one, in terms of like pure control, even if we directly feed the error back into the feedback control loop, that is proven to be, learning this new trajectory in the sense like, it's called iterative learning control, where you directly feed the error back into the feedback controller, and somehow the loop stabilizes into following the trajectory in similar number of iterations, like 400 or 300. So how could this be different than an iterative control scheme without any memory?
Sorry, I have not understand you.
Let's say without having a cerebellum, even if we directly compute the difference between desired and actual trajectory, and we feed it directly back into the original control loop,
yes,
without any cerebellum, that can reduce error also. I'm not sure if it can reduce it like very well or not. How could adding a cerebellum loop and be different from an iterative learning control scheme like this, where you directly feed error back into the control loop?
Well, in this case, we have a sensorimotor delay of 50 milliseconds between the control and the robot. If you try to control the robot with a position, with a PID, using this sensorimotor delay, it's almost impossible. You have overshooting, for example. We are studying now to control a robot using Wi-Fi connection. So we have the controlling a computer, and we are able to control a robot that is moving in a room with a Wi-Fi connection. This is not possible with a position control mechanism, with a PID, because not able to deal with these sensorimotor delays. It's, for instance, one advantage of the model.
I have a question myself. How many neurons do you have in a single module? Because you say you have 6 modules.
Yes.
But I don't remember, you have 7 degrees of freedom, but
yes,
only 6 modules.
Yes, because the last joint has some static problem, so we don't use.
You don't need,
you
don't use it, yes.
Because of some technical problem.
There are actually 6.
Yes.
6 degrees of freedom, 1 module per degree of freedom, that's understandable, but then you have how many neurons per module? Maybe, that's here. Sorry, I didn't get it.
6. So it's a simplification of the model. We don't have Golgi, molecular interneuron. This is a problem of computation. We just implement what we need, the minimum number. We will try to increase this model and include more neuron, more type of neuron, more a second plasticity mechanism in MOSI fiber to deep cerebral nuclei, but we will do in future work.
And the input from the MOSI fibers is a liquid state machine, basically?
No, we use Gaussian function to transfer the analog signal to the MOSI fiber activity.
Okay. Good. Thank you.
Okay.
If there are no other questions, then we are super late, but we can start with the next session, and we can try to do part of this last session now and part after lunch. That's the only way we can cope with it, because lunch will be in 15 minutes. So we can start with the last session, that is the session on the virtual mouse brain embedding cerebellar nodes. And then we will complete this session just after lunch. Okay. Okay, so as we did with the previous, some of the previous sections, I just give an introduction, I give a sort of overview of the section, and then others will continue and give all the elements that we need. So now we are at the virtual brain embedding cerebellar nodes. It is a sort of continuation of the robotic controllers embedding cerebellar nodes, because again, we have models of the cerebellum that is embedded into larger systems which have an architecture, have some rules, and then have, in the two different cases, two different applications. In one case, they are applied to robots, as we've seen. In the other case, to simulate brain waves. I play back this picture that was shown on the first day on how do we create brain models. There is a section that runs bottom up, is the section on the left, the reverse engineering section, in which the models are built starting from principle of cellular biophysics, going up to realistic neuronal models and realistic microcircuit models. At the moment, this is the best we can do. So we can make models starting from a large set of data coming from biology, passing through the rules of cellular biophysics, generating realistic models of the neurons that can actually represent what we know about the neurons. This can be connected into networks, generating local microcircuits. This can be quite big, tens of thousands or hundreds of thousands of neurons. They are computationally heavy, but biophysically and biologically very precise. So we are quite happy with these models. The point is that they are difficult to be used, hard to be used in the context of large-scale networks, long-lasting simulations, and applications to real-time robots. So the point is here to find a compromise between these models and the knowledge that we would like to achieve about the system. The system is normally investigated using top-down approaches, which are complementary. The top-down approaches starts from observations of the system as a whole, where we measure global parameters. For example, the waves generated by the brain and detected using an electroencephalogram, or the oscillations in the MRI signal picked up by the MRI scanner. So these are ensemble signals that do not see the single neurons, but see the ensemble contribution of many neurons. Moreover, there is another problem. Activity in the brain is organized in a complex architecture in which there are areas that we can identify quite well, areas that are activated with a specific meaning because they are related to function. This area is activated during motor control, the other area during vision, the other area during whatever. So basically, we have different areas activated in different tasks. So in order to cope with this complexity, neurons on the one hand and areas and functions on the other, we need to let the two approaches converge. How do they converge? The one idea is to use virtual brains. The story of the virtual brains, Spasse is here, and will be explained properly by Spasse. The point is that these virtual brains have nodes, and the nodes are representative of the local activity into the microcircuits. So the point is how to include the nodes, and the other point is how to represent and construct the nodes. This has to do, on the one hand, with theory, biology, biophysics, understanding the brain, but on the other hand, with technology. How do we do it? Because depending on how we make the node, the simulator is different. So we need to adapt the simulator to the kind of nodes that we use, or vice versa. We have to make the nodes to fit the simulator. So there is, as normally happens, a sort of ping pong between technology and science. So how do we make the nodes? A brute force approach would be to make spiking networks and put them into the simulator. This is something that goes under the name co-simulations. Is a sort of pioneering approach that has been proposed already in some test trials and that can be engineered further. Surely we would be happy to use it. In the meanwhile, it's important to have more synthetic representations of the nodes in terms of what we call the neural masses or mean fields. The neural masses and the mean fields are representations of what a complex set of neurons do under certain circumstances. For example, we give an input, the neural mass or the mean field will react, generating an output. And we go to scale, in a way, with the size of the areas that are detected by electroencephalography, magnetoencephalography, and functional MRI. These areas are much above the level of a single neuron. They include tens of thousands or hundreds of thousands of neurons. And therefore, what we see with a signal is more related to the mean field than to the single neuron. The step to move from one to the other, from the single neurons to the mean fields and the activities of an ensemble of neurons are complicated. It's not easy. It is something that is under investigation. We have to go back and forth. Going forward is, in a way, in principle, feasible. Going backward is a bad problem in mathematics because it is an inverse problem. We do not have the way to understand what the individual sources do in generating the ensemble signal, except with a very nice model in the middle. So again, using the models is critical. This is how the virtual brain strategy can be represented. I don't want to go further in this because Pase will do it. I want us to point out a couple of elements that we need to understand the problem. We have basically a central role for magnetic resonance imaging in generating the structure of the connections that we call edges, and in identifying through mapping the nodes that are used to reconnect the brain. Then what comes out is a structure like this that is made of nodes and edges, or nodes and connections. The nodes are the point where we will focus because, in particular, the nodes of the cerebellum have never been developed properly. So how are these nodes developed normally? As neural masses or mean fields, or spiking networks, in the best case, but more, I would say, hard case to apply. The neural masses are simplified representations of the ensemble neuronal activity. And this can be written down using equations that generates as an output waves. So it's an analog representation of the activity of a group of neurons. These waves can be communicated from one node to another node, generating a system of a coupled system. And since each node normally has the property of an oscillator, we have a system of coupled oscillators. So finally, the brain represented here is a system of coupled oscillators. This applies, in a way, to the cortex. The cortex has been represented in this way quite successfully, obtaining interesting information about how the cortex works and how the brain waves observed in the cortex are generated. What about the cerebellum? The cerebellum has an internal microcircuit that is very different from that of the cerebral cortex. It's based on different construction principles. And it doesn't easily fit with the representation of a node normally applied to the cerebral cortex. So we have to rebuild the neural mass of the cerebellum, or the mean field of the cerebellum. We have to make it anew. So this is an important point. The other issue is that the cerebellum is strongly connected to the cortex. So it's very hard for me to understand how one can represent the brain only using the cortex. It's not the brain, it's the cortex. Half of the neurons of the brain are in the cerebellum. Why should we lose them in our simulations? We need them. And there are millions of fibers traveling from the cerebellum to the cortex and back. The tractography that was done in these years, for example, by Fulvia and Claudia, this tractography shows very nicely that the cerebellum is connected to large areas and large sections of the cerebral cortex through the thalamus and back. So if we lose the cerebellum in the virtual brain, we lose a big part of the brain. This set of consideration prompts us to develop a strategy to first have virtual masses or mean fields working for the cerebellar node. So we have to design them and implement them, then to transplant the nodes into the virtual brain. So this is the operation. Spazen knows it well because, I mean, we have been discussing of this story starting a couple of months ago. And today, there will be the first assessment of the state of the art, or where we are, what we need to do, and where we want to go. Typically, this is a picture that I have already shown in my introductory lecture yesterday. We are going toward a neural mass. I mean, I would say I'm always mentioning both the neural mass and the mean field. Probably what we're doing is closer to a mean field than a neural mass because we are taking into account the spatial properties of the system. So we're taking into account the field properties, not the single point collapse of all the properties. So in this mean field representation, we have, I hope it works. No, it doesn't work here. In this mean field representation, we have maintained the properties of the network. These properties have been investigated during biological experiments, have been represented in models done with single neurons and models done with detailed representation of the neurons. But then these properties have been extracted as properties that can be written down as equations or embedded into the mean field. For example, this mean field maintains the capacity of working as an adaptive filter in space and time. Can filter information in space and time. This is a critical property for a cerebellar node. It maintains the property of oscillating, is an oscillator. And this is critical because we need oscillators inside the construction of the virtual brain, and the cerebellum is an oscillator. It has the property of working as a perceptron, and to reconnect the information coming from outside the cerebellum to perform operations that we have seen this morning, like spatial decorrelation. And this operation is maintained, and we have a perceptron recorrelating all the information. And then this is still to be done, and Claudia will tell something about it. We have at some step into this process, we would like to have a virtual mass in which we embed plasticity with internal rules. That's another step. It's not always done with the normal virtual brain construction as far as I know. So we need to have plasticity rules, allowing the cerebellum to learn and calibrate the weights inside the nodes. So this is all the story I wanted to present now and to summarize. And I also would like to mention that in order to do this job, we are involving all the laboratories of our group. And in this case, the neuroimaging laboratory is taking a critical part because most of the work is done in the neuroimaging laboratory in collaboration with the neurocomputation. I now leave space to you, and the next one is Pase, which will explain the main concepts about the virtual brain.
everyone. So I'm the last speaker before the lunch. And yes, as Egidio mentioned, I'm going to describe the large-scale brain network modeling in general, and especially how we are doing it with the virtual brain platform. So I'm coming from the Institute of Neuroscience, the system from Marseille. And since the work on the introducing thalamus in the TVB is still very much ongoing, I will speak now about how mice data can be, mice connectome data can be integrated in the TVB. And I will shortly present one use case which is more finished than the thalamus work, which is very much ongoing. While later in the tutorial, actually we'll have a few exercises about the thalamus connectivity and how that impacts the overall macroscopic dynamics of the brain. So the paradigm behind the large-scale brain network modeling, as also Egidio mentioned, is that we assume that the brain can be divided in a separate anatomical regions, and that we have a way to find the actual structural connectivities between these regions, how these regions communicate between each other. And then once we have this, then the overall activity that we measure, the macroscopic activity, no matter with what modality, what neuroimaging modality or electroencephalography, over the whole brain, the emergent dynamics is shaped by this network, by the connectivity of the brain. And what we usually then work on are these neural populations, or which then can be the activity of each region of the brain. Then we assume that if this brain region was taken apart from the brain, its activity, its mean activity, can be defined by some activity of a neural population, which then can be described by a neural mass or a mean field, as Egidio said, and we end up with the brain network model. And here I have a brain network of a human, and just to mention that currently in most of the models for human, we have only two nodes for the cerebellum, cerebellum left and cerebellum right, and it's the same case with the thalamus or with hippocampus. And I think the problems is that mainly the data that we use to validate the model, so either a functional MRI or a whole scalp EEG, or we work lots on epileptic patients, we cannot do a proper imaging on the thalamus, and that is one of the reasons why we don't represent it better. And then the other issue is, of course, that probably the neural mass or the neural population which is in the cortex then is not the same as the neural population which is in the cerebellum. And then in the TVB we have two levels of representation. So we can have brain nodes only, or we can have a neural field. So the local dynamics is about the neural field, and then we end up with like several thousands of edges or nodes. So usually it's the cortical sheet. These nodes are connected with some kernel, connectivity kernel, which is homotropic in space. But then, on top of that, between the different brain regions, we have the global dynamics from the connectome. And then the overall field potential, meaning the dynamics that one can expect to appear at each point of the brain in space and in time, it's actually defined by the interplay of the local dynamics, of the inherent dynamics, the local dynamics around that point in space, but also from the long-range projections, the axonal projections. So for this, we have the neuroinformatic platform, the virtual brain. For that, a bit later in the afternoon. Here, I'm just showing some screenshots that there is a quite useful graphic user interface, which is usually used by medical doctors, for example, but then for computational neuroscientists, it's written in Python, so there are very nice Jupyter Notebook tutorials for the beginners. We'll have one of those later. And then there is also a nice forum where you can ask any questions. Usually they're quite quick responding. There are interesting discussions. And now I will move on to how we can use the TVB, or large-scale brain network modeling paradigm, for the mice. And for the mice, I will focus on the data, which is now getting more and more used, which is, I guess, the best, the most detailed tracing connectome data for the mice, which comes from the Allen Institute. And with the finest resolution there, we end up with 540 different nodes of the brain. So we can define 540 nodes, and we have the connectivity matrix of 540 times 540. And I just had a look here. I knew about the cortex, so we either use the whole brain or the cortex, depending what is the problem that we are modeling on. The cortex has 86 regions, 43 per hemisphere, and the cerebellum has 32 nodes. So it's quite better represented than what we get from MRI imaging. And then from DTI and MRI in mice, I also think that one could not get more than few regions for the cerebellum. And then interesting, the brain stem is quite well represented. There are 360 regions. And now I will shortly describe the use case about the stroke. So basically what we are trying to model is what is the dynamics before and after the stroke. And when I say dynamics, it's the dynamics as seen by wide field calcium imaging. I will show it later. So basically we have a field of view. We have the animal when it's healthy, no stroke, and then stroke occurs in the right primary motor cortex, and we try to find what is the changes in the functional connectivity that we observe in the data, how much we can explain them by the rewiring of the connectome, because the stroke destroys some of the connections, and then we also have the results after 30 days of recovery on some special platform when it's assumed that also rewiring, structural rewiring occurs in the brain, and since we don't have the scans of the connectivity, the DTI of the different stages when the animal is healthy, then after stroke, then after a full recovery, we are trying computationally to predict how this process goes and which parts of the brain are mostly affected. And since we are looking at the phase coherence at a band that we have chosen that is of interest for the resting state, which is the lower delta frequency, we also use, for this use case, the Kuramoto oscillators to describe the oscillatory activity. So simply if we don't know any better, if we don't have more detailed information, and if the problem that we are describing, like in this case, is coactivation, synchronization on different brain regions, it's fine to use, like in this case, Kuramoto oscillators. But if we are interested, for example, in modeling how epilepsy propagates or some spikes, then we need a model that is able to describe that. And that's why in TVB one can choose different models depending on what question they are asking, what they are trying to model. So about the different structural connectivity, just to mention the main differences. So far we have either a tracer data or diffusion MRI. And the spatial resolution, it's much better in the tracing data. And then of course, it's better because it actually gets the actual links and the connectivity between different regions, while the diffusion MRI uses lots of processing, so it's not really exact direct link of the connectivity, but using how water molecules behave under the magnetic field, one can assume where are the white matter tracts. And then this can fail in the long and complex connectomes we don't have directionality. However, it's important because it's the only way we can get connectivity in humans, and that it's also individualized. So in order to get the full connectome, the connectome of the full brain using tracers, first of all, can be done in humans, and even when it's done in mice, it's done over many animals. So then again, it's some average connectome, and depending what we are interested in, it might be of importance to have the individualized or personalized connectome. And then I will demonstrate this later. So all this database from the Allen connectome, it's available in the Allen Connectivity Builder, which is part of the virtual brain. So one can choose the level of how coarse the connectome that we are importing we want to be, how many experiments per region, how reliable this to be, etc. And we end up with the structural connectivity with a different number of regions. And then this, so here we are shown two different, like coarser and finer, okay, this doesn't work, finer connectomes. And interesting, it was just published that we have validated the Allen connectome because, right, we have this data set, which is very detailed, but how can we be sure that this has anything with the reality, that it's useful? So basically, what we've done, there was a data set of, I think, around 15 mice from Israel, and then each of them had 4 to 5 resting state fMRI sessions. And for each of these animals, there was obtained the personalized DTI connectivity. And then we tried our best to model the observed resting state functional connectivity for each animal using their own connectome and using the Allen connectome and using different surrogates to see what is actually, why the one which is better, what is the reason. And like important is, for example, the first bar here showing the scan-rescan correlation. So for the same animal, in 2 different sessions, the correlation is around 0.6. So if we are trying to model now the functional connectivity, there is no point for our model to reach higher than 0.6, right? If 0.6 is the ground truth. If tomorrow the animal can have slightly different connectivity. So we should be aware about the bounds of the problem or how much we can simulate and to be careful not to over-simulate. And just the main point here is that, in fact, the original tracer data, which is the green bar, has better predictive power even than individual mice connectomes. So using this, the average connectivity from many mice from the Allen Institute, but which is the actual links, we get better predictability. And then, of course, if we have to use MRI, it was also shown that the individual MRI has higher predictability than the average fMRI of all these 15 mice. And then using different shufflings and surrogates, it was found that indeed it is this directionality in the tracer data, which is missing from the DTI MRI, and the complex fibers that are indeed causing the Allen connectome to be more predictable. And now, so because of that, and because we didn't, so here we are showing that Allen connectome is more predictable for resting state. It doesn't mean that it's more predictable for if we have a mice with a stroke or a mice with epilepsy. However, so the experiment that we have here, the data is from Florence, from Francesco Pavone's lab, and Letizia. So they had these several mice animals which got stroke, and then they spent some time rehabilitating. And then since we don't have for this the personalized DTI, if we had, it would be the best to model what is happening using their own connectons and the Allen database to see which one is better. Moreover, because we know that the stroke induces structural changes. So in the original database, I will say later, we can just computationally try different scenarios, which regions are affected by the stroke, and see which of these shows the best predictability of the data. So we have the stroke. That's how the data looks. So basically, it's wide field calcium recording, depending which animal it was, either one hemisphere or two hemispheres. We overlay it on top of that the division of the cortical regions that we have from the Allen Atlas. Also later I will show the Allen Atlas visualizer. And so depending on the experiment, it's either this field of view or only this side. And then we end up with, again, depending on the animal, 18 or 15 different regions. And then for the activity in these regions, we average the activity of the luminosity of all the pixels which are within, and we end up with the mean activity of all these of the regions, which looks something like this over time. So we see the high spikes where due to the movement that occurs on this platform. However, we are interested in modeling the resting state because this one is the most affected by the structure. And with the brain network modeling, we are interested how much the dynamics, the function is shaped by the structure. So that's why we are looking at the frequency band where we have really the resting state, not really artifacts, but where it's not the wave, the slow wave from the movement, but really the resting state. So for that, we are choosing the upper delta band, and we are then describing phase locking values between different regions and trying to see are there significant changes between stroke versus healthy state versus different levels of recovery. And what we end up, this is all from the data, is with connectivity matrices. So here are the different brain regions, and we see how they are connected between each other in different conditions, meaning healthy or stroke. And yes, it can be pure correlation or PLV, phase locking value at different frequencies, depending what is your paradigm, what are you testing. Then there was day by day variability, so we take the medians of this functional connectivity so that to end up with really the mean functional connectivity of control versus stroke. And this is actually not surprising, because if you remember what I showed, day-to-day scanning, even for healthy animals, it's 0.6 from this other data set that we used to validate the Allen connectome. And we end up with some matrices for changes of the connectivity. So how much connectivity? So week 1 is the stroke, so just after the stroke, and week 4 is after rehabilitation, because really these animals, after a month of rehabilitation on the special platform, they are moving almost perfectly, and there is no, they look like there was no stroke. But we are then looking what is in the data, what are the specific functional connectivities typical for the control and for the stroke versus control and for the recovery versus control. And then using this, we are now modeling different scenarios. So we are building the mouse brain network models, meaning we have now the network nodes, which I showed in the beginning for the cortex. In each of them, we put one oscillator. And then depending on the different levels of the global coupling, which says how strongly connected is the overall level. We cannot change the connectivity at each region, but overall level, and then the rest is shaped by the connectome. And we are modulating different levels of the stroke impact, saying what would happen if only 10% of all the links to the primary motor cortex are affected, or 20, or 30, or what if this region is completely disconnected from the rest of the brain. So then we test all these scenarios and we see which one of them will later give the best predictability or the best correlation with the data. And the other thing that we analyze is the rewiring due to rehabilitation, because not just the impact of the stroke when the links are removed, but there's also rewiring between other regions, especially like contralateral regions, which then probably affect both the dynamics and the recovery. So we do the same processing and metrics for the data as for the model. So we end up with some changes of the functional connectivity in the control versus the stroke in the empirical data and in the model. The details are not important, so we end up really in this parameter space with a different levels of model predictability depending on how strong is the stroke and the rewiring. These two are the parameters that are important from structural point of view to describe these three different conditions. And indeed, the model shows that there is much better predictability, so going even up to 0.6 or 0.7, if we have higher damage for the week 1 and smaller rewiring than at the week 4. And at the end, we also did statistical significance testing. So yes, if we shuffle this Allen connectome, then you end up with the correlation around 0 and with Gaussian distribution. So yes, indeed, there is some information in the structural connectivity. And then by testing different scenarios, we can try to predict which one indeed happened. With the particular stroke, which connectivities were the most affected, and then with the recovery, where it happened that we got the most new connections or rewirings. So the summary that indeed, yes, there is, we've created a brain network model. So using the cables, it's time to go. But yeah, just the summary that, yes, the large-scale brain network modeling can help us to find the actual impact of the stroke and the recovery. And there are, of course, some open questions, such as with the stroke, there is shrinkage of the tissue, but these are not important for here. And yeah, that's all for now. I want to thank my collaborators and thank you for your attention. Questions?
Questions? Claudia?
Sorry, one question. When you spoke about, let's say, local connectivity to within a node, I understand it's like a sort of spatial kernel, but connected what? Something like, I mean, inside a node, you can impose this local connectivity, right?
So then the same node that would be again on the brain network now goes on the edge. So instead of network of 100 nodes, you end up with network of 10,000 nodes. But then the connectivity will be, each of the nodes will be with some kernel connected to its neighbors. And then there is a long-range projection
from the global.
Shown here from the connectome. Now, of course, there is issue to find the right balance, which one is stronger, the local or the...
So you have a sort of some effects from the local and then the global long-range ones. Okay. Thank you.
They are, for now, when we don't know any better, they are all identical. So, for example, for epilepsy for human, or for mice, we have a special model for the seizure propagation and so on. So from the data from the clinicians, then we can have information that in this region there is higher excitability. So we put that in the model. So even here, we can put different, we can put a heat map, spatial heat map with different parameters. So either the same model can be different for the cerebellum, in this case, or it can be a different model. But this needs to be, there should be good evidence for this.
For that. To introduce this edited feature, let's say, into the connection.
Yes.
Okay. Thank you.
But in the simulation that you're doing with the virtual mouse brain now, I understand that this is done on the Allen for the connectivity.
Yes.
And on data from Francesco
from
for
the
calcium imaging in mice. Is there any cerebellum inside there?
In the model, here what I showed, we used only the cortex. So we used either only cortex, 86 nodes, or the whole brain, 540 nodes.
Whole brain.
And the whole brain also contains 32 nodes of the cerebellum. So, but these nodes are only for the connectivity. From the functional data that we use for the validation, of course, it's only cortical regions that can be seen by the camera. But still in the model, and that is also later in the tutorial, even the connectivity on the cortex, of course, it's very much different depending on, is it only cortex, or is it whole brain, or cortex and cerebellum, or cortex and cerebellum and thalamus?
Okay, thank you. Now we will have time to talk of this later.
Okay.
No more questions? One question.
Hi. I was wondering if you were planning to integrate the new AAV tracers injections from the Allen Brain Institute from the Harris paper, for instance, in 2018, which were more precise for the cortex. And a second question related to it is for some regions like the cerebellum, you have very few injections, which are not covering the entire cerebellum then. So what's your plan to compensate for that?
So for the first question, I think even now in the TVB builder, it would be, if one imports the connectome, it would be the new data with the several layers for the cortical regions. For this model, it's not necessarily because of the level, because of the imaging data of the calcium imaging, so it wouldn't make us many difference, but if there is some, if your data would be then implanted electrodes at different depths, indeed this would be very helpful. And for the thalamus, yeah, I know that issue, but then again, same like with the MRI, we use what we have. So if that is the best connectome now that there is, we would use it. For sure, it's still not perfect. Hopefully they will, get better connectivity, or then maybe this can be all resolved with a neural mass, a mean field of the whole thalamus, which then takes into account different properties, and then maybe this one region can be more helpful if it's done a different neural mass which properly describes it, than having 20 or 30 or 50 regions.
Okay, if there are no more questions, we move up for lunch. Thank you. Thank you. Now 2 presentations to complete the section on the virtual brain. And Fulvia first, and then Claudia. They promise to be short. And then we will have to finish the presentation of Armando. Armando is around here. Where is Armando?
He's coming.
He's coming, yeah, because Armando didn't have time to conclude the presentation yesterday. He will take, he says, 15 minutes to finish. Okay.
Okay. Good afternoon, everyone. I will be very short, and I will do a sort of recap of TVB. In particular, I will speak about the human brain structural connectivity and the simulation of human brain dynamics. As you have seen in the preview presentation, in order to simulate functional activity of the brain, we need essentially 2 different things. So we have to parcellate, divide the entire brain, the cortex of the brain, in different regions, and these regions are called nodes, generally are called nodes. The main difference between this kind of parcellation, this kind of anatomical subdivision with the other microscopic and detailed or simplified model is the spatial resolution. Here, each region has a volume or an area that is around some millimeters. And the second thing that we need in order to create the brain structural connectivity is the connection. So we have to connect different and distinct parts of the brain. And since we are working on humans, we cannot use some kind of tracing approaches, but the only thing that we can do is use some macroscopic probing, and one of the most used is the magnetic resonance imaging. In particular, here we use the diffusion imaging, diffusion MRI, and tractography to connect different parts of the brain. In order to define the nodes, generally, you can just identify the different brain regions, and what most of the studies have done until now is just consider what happens in the cortical area, in the cerebral cortical area of the brain, and in the thalamus, hippocampi, and basal ganglia, so the deep gray matter structure of the brain. But what we are now doing is adding the, not fine, but yes, a parcellation, a subdivision also of the cerebellar cortex. And in our works, usually we use 40 different cerebellar nodes. Okay, in order to connect different regions, we use the tractography. This is a very nice technique, but still has some limitations. The first thing is that, yes, is always the spatial resolution. We cannot reconstruct, we cannot connect microscopic distant area of the brain, but we just can connect some macroscopic areas, and so we cannot, for us, a connection is not defined as a single axons that connect to distinct regions, but is defined as a coherent bundle of several axonal fibers. And what is challenging is the reconstruction of cerebellar tracts and cerebrocerebellar tracts. Maybe this is one of the reasons, because the cerebellum isn't used and isn't included in the previous studies. Now, this kind of representation, so the whole brain structural connectivity, can be collapsed in a matrix that is the connectome matrix that Spase said about that before. And in particular, here we can define and identify different row and different column. Each column is a different brain area, while each square, so I mean each different color inside the matrix, represents an edge, so a connection, a structural connection. The important thing is that we can add a weight to each connection. So we can say not only that two different regions, brain regions, are connected to each other, but that they are more connected. The connection is stronger than another kind of connection. Okay, and in particular, when we are working with TVB, we usually have to define three different matrices, three different kind of weights. The first one is the normalized number of axonal fibers. So indeed, we can define the weight, how strong is a connection. Then we have to define the axonal bundle length, and this information is important in order to understand and define what is the conduction speed, which is the time of the conduction between distinct regions. And the final weight is referring to functional connectivity, to functional connectome. Here we use the Pearson correlation coefficient in order to understand which areas of the brain are functionally connected to each other. Okay, here Egidio has already shown this slide. Here is reported the general pipeline of a macroscopic, of a whole brain dynamic simulation. We can see that there are several steps of preprocessing regarding diffusion and functional images. After that, we can define the whole-brain connection, the whole-brain connectivity, and we can use this information inside the TVB in order to simulate functional and neuronal activity. Each node will have a distinct, separate neural activity. And using this information, using these waves, we can also create a connectome of the simulated functional connectivity. And this is what we'll do during the tutorial this afternoon. The final step is the optimization. I mean, we can just simulate a functional behavior of the brain, but we also want to know if our simulation is a good simulation or no. So we have to use also some empirical functional data in order to optimize our simulation and obtain the higher correlation, the best results that we can have obtained. And the other important thing is that this kind of pipeline, this kind of simulation, as Spase said, can be done at single subject level. So each different subject has a different connectome, has a different simulation, has a different functional dynamics. Now, it's important not only to understand how the functional dynamics are in the whole brain, but it's important to remember that some local brain dynamics exist, and in particular different different structures of the brain do different things. So what we want to study is not only the global brain dynamics, but also the local brain dynamics. And in particular, here are reported two different studies that were focused, the first one on the limbic subnetwork, and the second one on the cerebellar connectivity. Here is reported, results regarding the first two matrices. The matrices at the left are the input of the TVB. They contain information, the structural information, while the matrix on the right represents the simulated functional connectivity, so the simulated functionality of the brain. And in particular, in this case, we were only interested to cerebrocortical connectivity. Here is what happened when we added also the cerebellar connectivity connections, and in the orange square you can see what is the results of adding cerebellar connectivity. What we found is that the final correlation between the simulated and the empirical functional connectivity is not significantly changed when we add the cerebellum. And maybe this means that it's really important to have a good representation of the structural anatomy, connectivity of the entire brain. Nevertheless, it's also very important to use different models and different neural masses, that are specific to different parts of the brain. And this is the reason for which we need the introduction of a cerebellar neural mass. This is because cerebellar microcircuits organization is really different from the cerebral cortex organization, and in particular we have to introduce this kind of neural mass, this kind of new model, in order to account microscale phenomenon. And here is just a representation, but Claudia will talk about that. The last thing that I want to say is that what we expect by the introduction not only of cerebellar nodes but also a specific cerebellar neural mass in TVB is that simulation will be driven by a detailed microscale models, and so we should have better association between functional activation and cellular activity. We can investigate the spatiotemporal dynamics not only from a global point of view, but also from a local point of view. And in particular, we think that this can further improve our understanding of pathological diseases. And then Claudia on the stage.
Just a few slides. It's something already Egidio and Fulvia said a little bit, but so the idea is to have a more detailed, fine-grained cerebellar nodes description with the idea that, of course, the cerebellar has all the brain structure, but the structural properties shape strongly the processing. So what we want to try to keep its most as possible features within the description of these nodes, specifically for the cerebellum in this case, in order to really generate a cerebellar processing to have a powerful tool to understand the underlying mechanism of functional phenomena we can observe in the virtual brain simulation. So Spazi already explained the approach. So the idea is to lower the scale, so from a mesoscopic description of population behavior, we want to introduce some features we think they are crucial for the functional of cerebellar nodes. So we want to have a description of the node in a higher spatial sampling and create a local connectivity with specific geometrical features. So we have especially modules interconnected through parallel fibers which run in a specific direction of the geometry of the map, and this is it's important to keep the functional interaction among these modules. So we want to create a specific kernel of local connectivity based on parallel fiber features, so the direction, the length of these fibers, and also the input to these nodes. So we would like to introduce a sort of model properties on the global connectivity, so to create a branching of these fibers which reproduce important features like signal expansion or recombination into the different parts of the nodes. For instance, this is a node of the cerebellum, and we would like to build it as a combination of vertex connected by intranode local connectivity. So we need to create a system of equations, try to keep the dynamic properties of these features, so spatial and temporal properties. So we have units representing a granular layer, which works as centers around spatial kernel with a temporal dynamics, of course. So we have written equation of a Mexican nuts reproducing a centers around shape and a classical differential equation with temporal decays. Then we have a spatial integration of all these units, and also we introduced a big, let's say, Golgi cell, which create a synchronized inhibition on these input units. Then we have the perceptron, so the Purkinje cell collecting all these inputs processed by this array, and we have a unit of molecular interneuron inhibiting this perceptron activity in order to modulate this activity. So the idea is to have multiple vertex. The parallel fiber have specific weights from one vertex to another, keeping, of course, a geometrical mapping of these fibers, so creating extension and direction of this multiple vertex connection. So this is just an example of some input signal provided to multiple vertex. Then we have the Golgi cells of the two vertex, in some cases reaching a threshold and creating a synchronized inhibition. In another case, the excitation is not enough to have this inhibition on the underlying units. And then we have a sort of output waves coming from the activity of the perceptron. So this is just a very general design. We would like to, with the ideas to put microscopic features into this microscopic system, and then to have this abstract description fine grain of the cerebellar nodes, and to compare it also with a co-simulation between pi-nest single cell networks mapped on this node, running within the virtual brain compared to this abstract description of the nodes, but try to keep the same structural and then functional properties. So this is, let's say, it's the goal of next work in the SJ3 of human brain. So working with the virtual brain, trying to have a customized description of these nodes, and also have different tools running together and try to compare the results and the impacts of these features described with spiking single neurons, with abstract equations, but still keeping all the hierarchy of the connections and the spatial properties of these connections. Thank you very much.
So thank you, Fulvia and Claudia, for the presentations. Are there any questions on your parts? Alessandra?
Claudia, sorry. I mean, it's amazing the way you are thinking how to use the virtual brain into and keeping the cerebellar circuitry somehow condensed. When you talk about vertex, is the same as node, right?
Sorry?
Vertex and node?
No, actually what we try, but we should also understand
sorry.
better with SPAS. No, the idea is that the vertex is fine grain, it's a higher resolution with respect to nodes. So the nodes could be composed by multiple vertex connected by an intranode connectivity somehow.
So they are kind of local nodes. Okay, yes. But they are not the weights. They are not the edges. They are like nodes.
No.
And so you are associating the vertex with a sort of spatial local microcircuits of the whole population of the cerebellum.
Yes,
right? Cioè inside the vertex you have granule, malai, Purkinje, and
yes,
all different cells. So you are not associating a vertex with the specific cell population.
No, the vertex is, okay, as we have designed, but still we have to understand the feasibility to put, because the vertex right now, I think, Spase, correct me, it's more a geometrical map. It's not really, the vertex now is not maybe something with specific equation inside. It's more like a map of the geometry of the nodes, creating a local connectivity within the node. But what we would like to do is to as you said, to have a functional module in the vertex. So, like a multiple module architecture within the node, and each vertex, right, contains all the main circuits of the cerebellum.
But, sorry, at the end of the day, each vertex is a black box model. It has input, output, and an equation inside.
Yeah, it's an equation, system of equations.
Okay.
And with the local connectivity, which is more than the global ones, trying to, so not just from data set, but we need to put some model hypotheses. And the idea is that you branch the input to the node into the different vertices, knowing some principle of branching and connection among vertex within the nodes. This is the idea. So you have the data from tractography, anyway, long range, and then that one can be split in the different multiple vertex within the same node. This is the desire, but okay. We don't know yet exactly how to implement everything.
Thank you.
Okay, thank you. Okay, if no more questions, I think we should give some time to Armando finish. Armando, you're ready to?
Okay, so on Monday I presented the hippocampus model. For the people that couldn't attend, I work at a Blueprint project on the hippocampus network modeling. And so I describe on Monday how I introduce how we build the model. So this is the result. And then let's simulate it. So we use two simulators. Both are based on neuron simulation environment. One is called Neurodamus, and another one is CoreNeuron. Neurodamus is not available, but CoreNeuron is on GitHub, and the associated paper was published last year. Actually, I'm now using exclusively CoreNeuron because it really optimizes the memory and the cores. Because this network, consider that this network is pretty complicated and contains 400,000 neurons. Each neuron has roughly 1,000 compartments, plus billions of synapses. So I have to say, I haven't had much experience in simulating this huge network because we spent 5 years to optimize it, the anatomy, but then we just started to see what it does. So let's say we have 3 blocks of simulation. So the first set of simulation, just test simulation. So we want to see if the network behaves as expected. And you imagine that even if you are happy with the building blocks, so let's say the single cells, the synapses, and so on, it's not guaranteed that when you assemble all together, works as you expected. Just an example that may be interesting. So the previous version looks great. I mean, the single cell, synapses, everything looks great. But then when we assemble, it was super excitable, the network. So what happened is that when we tune the single cell, since our data were mainly on the soma, we actually tuned the soma. And we didn't put too much attention on the dendrites. So what happened is that when we assembled the network, the dendrites were so active that even one single synaptic input on the dendrite made the cell fire. So it means even if the network with spontaneous activity without any input was so active that cells were firing at 100 Hz, let's say. That is absurd in the hippocampus. So test simulations. Then the second set of simulation, just to see, I mean, what kind of regime the network can go. Oscillatory regime, chaotic regimes, silent, and so on. And then we can address specific scientific questions. So I would skip this last. I will show you something on the first 2 points. As I said, so we haven't explored yet enough the network to tell you a nice story. But maybe some ideas could be reused for your work. So here what you see different cell types. Don't pay attention to the name. But then here you see so let's start from this point. So you see that basically without any, so the network, if you switch on the network, is basically silent, okay? That's good, because as I said, in the previous version, it was already too excitable, even without any input. So this is a test simulation. Here what I did, I switch on spontaneous synaptic activity. So you probably know, I mean, synapses can spontaneously be activated, even without a spike from the presynaptic terminal. Just random synaptic input. So they can, I mean, the rate is very low. Normally, it's at 0.1 hertz, let's say, just to give you the order of magnitude. But considering we have thousands of synapses per neuron, they can still make the cells fire from time to time. So here I have all the minis activated, and you see the percentage, I mean, the firing rates, it's very low. Here I activated different populations. So this is a test simulation to see that if I switch on minis in different population of synapses, then the network became more and more active. Very simple. So this is another simulation that is all the minis activated, as you can see from the traces. There is a little bit of subthreshold activity. From time to time here you have a spike. The overall activity, as I said, is very low. But then, I mean, it's good really to look at the traces. So, for example, this traces that is flat, it's reasonable. So we expected this because it actually doesn't receive, is a cell that doesn't receive input from external innervation, it doesn't receive input from internal connectivity. So it's almost silent in most of the cases. Then other things to notice that we skip the initial transient. So normally when you switch on the network, you may have an initial transient. How long is? You have to check. You have to check when the network go to plateau, go to a stable state. Sometimes it lasts for a few hundred milliseconds, sometimes it may last a few seconds. Other things to notice, from time to time, well, in somatosensory cortex we had this problem, not so much in the hippocampus model, but sometimes the cells get stuck. It means under a certain activation pattern, they go, for example, to 0 volt or to minus 30 volt, and they stay there, even without input. It's like you have sort of another stable point in the single cell model. So we have tools to detect those kind of stuck cells, we call, during the simulation. So this is another simulation that was done in the somatosensory cortex, and I reproduce in the hippocampus. Here we are in the second set of simulation. So here we want to understand what kind of regime the network can explore. So on the x-axis, you see we scan the calcium level. So we go to 2.0 millimolar, that is a normal in vitro-like calcium conditions, to 1.2 millimolar, that is more in vivo-like calcium condition. Of course, I mean, you can imagine that more you move towards high calcium, the activity is expected to be bigger because it basically increases the synapse release. Then here, what we have depolarization. This is in percentage because it's the percentage of, so we inject current as a percentage of the current necessary to bring the cells to the voltage, the spike threshold. So we calculate the spike threshold is a certain amount of ampere, and then we inject exactly this current or lower or more than this. And again, you expected that at 75 there is not enough current to bring the cells to spike, but then if you go to 120, the cell spikes. And here you see, I mean, there are different kind of conditions in which or the network is silent, or the network show chaotic behavior, or there is something like epileptic-like behavior. And we can calculate the spike-spike correlation to see if there are correlation between cells. This is somatosensory cortex. I did the same in the hippocampus. In the hippocampus, the network is pretty silent, but I have to say I switch off the minis. Instead, in the somatosensory cortex, they have the minis on because I want to have a clearer understanding of the system. And then I will add minis afterwards. This is what we can do. I mean, in silico, we can do all these tricks to understand the system. This is, anyway, so overall, the network is pretty silent, so we need really to drive the network. This is another simulation campaign that we had. So if you remember from Monday, the network is a CA1, is a subfield of the hippocampus, and we have projection from CA3. Actually, the CA3 have a very heavy projection on the CA1, so they made a lot of synapses, let's say 20,000 per pyramidal cells and 1,500 per interneurons. So here I'm just scanning parameters, give a Poisson-like input from CS3 and see how the network reacts. Again, the idea is to see if there are some regimes that is interesting or not, and what kind of regime. This is all, and here I plot only the distribution of the firing rates of pyramidal cells here and interneurons here. In the next slide, you see the same thing, but here I plot a little bit the evolution over time. So here you have a raster plot in blue and the firing rates over time in red. So you see in some cases it appears sort of oscillatory behavior, but I mean, we have to, of course, push this further to understand if it's real oscillatory or just something that happens. So we need really to study the oscillation. Okay, so as I said on Monday, the code will be available in a couple of months. We are going to have a MOOC on EDX on the hippocampus analysis and simulation, so it's not available, unfortunately, now. I can give you a little bit of preview of what the analysis will look like, just to give you a feeling. So I open Jupyter Notebook. Okay, let's see if it's here. Okay. So this is a kind of analysis I normally run. So when I run a simulation, I have this simulation report, I call. Just it gives me an idea what's going on. I mean, if the network is silent, is not silent, what are the cells that are more active, less active, if it has some structure or not. So for each cell type, I plot, this is a raster plot, so all the spike times of the cells. Here I have the instantaneous firing rate is basically a binning of the raster plot divided by the bin size. This is an interspike interval histogram. This is a frequency plot, frequency histogram to see if there is a broad, narrow distribution of the firing rates. And those are some sample traces. Okay, I will show you a little bit of logic how I produce those. Try to ignore all this kind of messy code. So I will highlight you the main point. So we can create with BluePy, this tool we are going to release on March, a simulation object. The simulation object contains basically all the information that you need for the circuit, so we can go deeper and look at the circuit, the anatomical property, but also we can look at the simulation. So for example, with this, we can take all the spikes. Yes, sorry. Better? Okay. So for example, these you can query all the spike times. Normally the function, you have to pass the GID, so a reference for each cell type, T-start, T-end, if you want to filter by the time. And of course, I mean, with this information, you can have custom function that calculate the frequency like this, and then eventually plot the frequency. So normally the custom function doesn't require many lines of code. So here is basically the core of the plot. So I iterate over all the cell types. I query the IDs. And then I have already built-in function that plot, for example, the raster. Again, I have to pass GID. I have to pass the time start, time end, and a sample number of neurons. In this case, I say none, because I say I want all of them. And then I have, for example, firing rate histogram already is built in, and so on. So and then voltage collage give me the trace. Yes, so as far as, well, of course, as far as the network is in the either Sonata format, so we started with a BBP custom format, but we are moving to a more general format that we are trying to achieve with the Allen Brain Institute, called Sonata format. So we are trying to converge into this. It should be quite general, it should be quite, so can be adopted by all possible networks. If it's in this format, then the tool can be used to query the necessary information. Well, anyway, so this is a little bit, I hope I give you a bit of feeling of how the tool look like. It's very easy to use. And yeah, keep an eye on ADX and see when the MOOC will be available. Okay, any questions?
There are questions for this last presentation? Okay, if it is all clear, thank you, Armando.
Thank you.
And we may move on to the tutorials now.